health systems. So my group, Healthy Machine Learning at MIT, focuses on creating actionable insights in human health. And we focus on 3 broad areas, trying to improve models so that they can be more robust, private, and fair. And these are things that might be useful in any setting, but they're really inspired by the need for these items before we deploy machine learning methods in healthcare. Using state-of-the-art systems to audit healthcare and understand what makes processes run by humans, but maybe aided by AI advice healthy. And finally, understanding how humans can actually use AI in practice, because even the best model trained to be robust, private, and fair, and applied to a really fantastic healthcare setting, if used improperly, will not induce any benefits in society. So first, let me give you a sense of, I think where we're at. And where we're at is pretty good with AI in health. So if we have a patient like Sumana who's having trouble breathing, and we want more efficiency, more efficacy with the system when lots of patients come into the intensive care unit, we might, for example, observe a lot of patient data, and a clinician could collaborate with a machine learning researcher and say, I want real-time prediction of something. So for example, this patient needs a ventilator, this patient needs a vasopressor. And by observing tens of thousands of labeled examples of when people who had signals like this in the intensive care unit needed these treatments, could you predict it in real time before some gap time that they would have needed it, 6 hours ahead of time, something like that? And so what a machine learning person like myself might do is take tens of thousands of records from a real hospital, a real intensive care unit, take all of the data, because we can do that now. We can take static data that does not change over the course of a stay. We can take time varying vitals and labs. We can take clinical notes and embed those using transformers, BERT models, any sort of system you like. And now everything about a patient over time in a hospital can be represented as a matrix of numbers. And if we have one matrix of numbers for every person, we then have a tensor of data over all the patients in the hospital. And then there's really great methods that can be applied to large tensors of data to notice patterns, and notably many neural methods. And so we found, this paper was several years ago now, one of the first that I did using neural networks in healthcare, that using neural network models in these settings, you can actually predict many different kinds of hospital interventions, like invasive and non-invasive ventilators, the need for a vasopressor, a drug that ups your blood pressure, or different kinds of fluid boluses, which much better performance than we thought possible, far ahead of time. So where the clinicians would actually want some sort of notice so that they could increase staffing. But, I'm not the only person who does this. There's a huge community of people who work on machine learning in health. And there's a range of tasks across the human lifespan where clinical AI has been demonstrated to perform at or above the humans it's been benchmarked to. But the question is, where will we go from here? Because machine learning researchers are really, really engaged in this area. It's a topic area that not only has a sort of submission category in the major machine learning conferences, you now have specific machine learning for health conferences all on their own that you can submit work to. And models are a regulated advice giver, right? And so the FDA actually recently announced that they're going to update rules so that you can have models retrained after they've been deployed, as long as you follow some specified retraining and deployment plan. But what are these models learning? So maybe you've been to the doctor recently. Maybe the doctor was tired or busy or distracted or just made a mistake, and it was corrected, everything was fine. But that data is now in a medical record system. And if you learn from all the data in a medical record system, you're learning from mistakes. And so one of the things that I'll focus on in this talk is we need more access to larger amounts of diverse data. Obviously, there are things that you have to consider when you're talking about health data in different jurisdictions. But if we want AI to actually improve science and address medical harms, we need diverse data sets for research use. And in general, right now, as it stands, health really lags the other machine learning subfields in terms of reproducibility. And so there's a huge push in areas like vision and natural language processing, and just generally in machine learning, to have code releases, data releases, and make sure that you're leveraging multiple data sets. But there isn't quite the same culture in health yet. We're getting there, but we still have a lot of work to go. So now that I've convinced you that you can do fantastic things, and hopefully it's not just me that's convinced you, you've looked around, you've seen the news, you can do truly fantastic things with machine learning in health. The question is, should you, and in what settings? So let's take an example. Let's say that you're a busy hospital and you just want to do triage with AI. So you're going to take the 3 largest chest X-ray data sets that exist, that are publicly available. That's over 700,000 images. And you're going to train a dense net, a kind of convolutional neural network, to predict no finding. The patient is healthy. I looked at their chest X-ray. They don't need to wait in the emergency room for the next 4 hours to see a doctor. They can go home. And we're going to compare the false positive rate of a state-of-the-art model, a model that would be put in that nice big table that we saw that performs at or above clinicians on average in this task. And we're going to call the false positive rate in different subgroups the underdiagnosis rate. Why would I call false positive underdiagnosis? Because if you falsely positively say somebody is healthy, they're actually sick, and you're going to send them home when they should have received care. And if, for example, your false positive rate is higher in one subpopulation, like female patients, it would lead to a higher rate of no treatment for female patients that actually need it if the model were deployed. So we run this to get the best model possible, outperforms doctors on average, and find that this state-of-the-art convolutional neural network model has the largest underdiagnosis rates in female patients, young patients, Black patients, and patients on Medicaid insurance. And intersectional identities are often more underdiagnosed than your aggregated group. So that means Black and Hispanic female patients are more underdiagnosed than white female patients, or than female patients in general. And you might say, like one reviewer did to me, that's fine, you're making a very simple point. We should just audit our models. Maybe we should just find a few predefined categories, and we should, tell the FDA, hey, pick your categories, make sure that every model before it's cleared reports a performance like this, so that we know, for example, that there's a difference between performance in public and private insurance. But it's not always that simple. Maybe you've heard of transformers, right? Language models are all the rage right now. So what happens when you take a language model, in this case, Cybert, that's only been trained on scientific articles? So it hasn't been trained on the internet, where you might imagine there's very problematic content. It's trained on science, scientific articles, that should be fine, right? And we took a real snippet from a patient note. Blank patient became belligerent and violent, they were sent to. And this is a real application. Doctors right now are actually able to use with many services. There's several startups that are offering as a service note completion in a medical setting using services like GPT-4. But if I start the note with Caucasian patient became belligerent and violent, or white patient became belligerent and violent, Cybert, Scientific BERT, fills in the rest of this patient's medical note with they were sent to the hospital. If I say African, African-American, or Black patient, same context, became belligerent and violent, those patients, their note gets completed with they were sent to prison. This is really problematic because these services are being used right now to automate the efficiency and efficacy of healthcare processes. And some people I've spoken to have said, maybe this is a right now problem because the data we have right now that we're building all these representations with are biased. But we have to make some progress right now, even if it's biased, even if there are gaps. Later, we'll have downstream classifiers with perfectly fair data, and that'll fix this issue. We actually ran that study. It turns out if you have a representation that you train, shown here by a nice sphere, and then you have a classifier you train that uses that representation, even if you have perfectly fair balanced data at the classification task level, if you have a biased representation because you learned a long time ago on biased data for your model, it's going to propagate to the downstream tasks. And so this is not something we can kick down the road and say, eventually we'll have perfect data and we'll be able to use these representations, but we'll balance them somehow. This is a problem we need to think about right now. Because bias is a strong part of the clinical landscape because doctors are humans and humans are biased. So we're going to see societal biases in healthcare data. How biased could it be, you might be asking? So pop quiz number one, I hope you're awake on this Friday afternoon. This is a nursing progress note from an area hospital where we've stripped out all of the self-reported patient race. Can you tell the race of this patient? Self-reported, nothing biological here, no genetic ancestry. So doctors couldn't, we've tested them, but machine learning models can. And here, for example, maybe the model is using things, spurious correlations that feel fair, right? So in the Northeast, where this research study was done, the two hospitals both have a higher proportion of patients who need dialysis and have hypertension who are also Black. But we found that this is not true for only very specific cases like this. If we look at two different hospital data sets in the Northeast, one in Boston and one in New York, this is over 4 million clinical notes across the patients in the two sites. We found that it's possible to predict self-reported race from clinical notes with a very, very high performance. And that's when using very simple models like logistic regression. And obviously it gets much better when you use language models like transformers. And the problem here is that some of these things like comorbidities, again, they feel fair. Maybe it's just so happens the model is cheating with the spurious correlation because it really wants to get it right. And it's noticed more Black patients need dialysis. But we've also found things like, for example, if you reference the patient's skin at all, that patient is probably white. And this is a well-known problem, even in dermatology textbooks, where there are very, very few examples of skin-based presentation of illness in patients with darker skin. And so what this is translated to is doctors often miss the skin-based presentation of illness or don't know how to recognize illnesses in darker skin patients. We also find some very personal things that the model's picking up on. If you ever use difficult in your note, that patient is probably going to be Black. And the problem here is that we thought initially, maybe they're saying the cases are difficult. Maybe we're saying it's a difficult stick. The patient is difficult to treat. In general, that is not universally true. People are making subjective assessments of Black patients and their families as being also just subjectively more difficult. And maybe you can see how my personal biases might creep into exactly the detailed ways that I write a note. And then, a machine learning model trained on millions and millions of examples would notice these patterns over time. What about medical imaging? Surely this is safe. Is this patient Black? Remember, this is not genetic ancestry. This is just self-reported race. So we find that, again, radiologists cannot tell somebody's self-reported race from a chest X-ray, but models can. And they can across many different sites, even when those sites are evenly balanced at every university, for example, for African-American and white patients. We also find that this is true across many different imaging modalities. And I just told you the secret of like, hey, maybe it's, these proxies or spurious correlations that the model's cheating with and using to figure out somebody's self-reported race. Let's figure out if that's true here. And so we tested the most obvious ones in this paper. Radiologist said maybe it's using BMI. It's not. Is it using breast density? No. Is it using bone density? No. Is it using disease distribution? No. In fact, this is probably fundamental to the imaging modality because we find that even when you bandpass filter these images to where they don't really look like chest X-rays that much anymore, you can still tell with really, really good performance whether somebody's self-reported race. A more melaninated person probably is more correlated with self-reporting African-American race. And the calibration of these medical imaging devices is likely giving some spectral information about that. And so is this bad all by itself? No. But if a machine learning model can tell perfectly that somebody's self-reported race is Black or white, and a clinician, a radiologist, who often doesn't they just get the chest X-ray and maybe some history, but they don't get the patient's self-reported race, cannot. And a model tends to be biased, maybe intentionally or unintentionally, against one category of person. The clinician would never know. because the model has access to information. It can infer something about the patient that we cannot. And so I think one of the things that is very common at this point, in a conversation with my fellow machine learning computer science people, is they say, hey, this is really a problem with black boxes, right? That's the actual problem here. Can't we just use explanation methods? Because that'll help us fix this. And so we actually decided to test this. So we thought, okay, let's actually evaluate these post hoc explanation methods, so both local models and global explanation models. And just for reference, if you are not in this space, these are extremely popular. The Lyme model alone has over 8,000 citations for post hoc explanation of a black box model. And then we're going to measure whether these explanation methods, right? You have a big black box model that has some performance with some kind of accuracy in different subgroups or other metric within different subgroups. Once you explain it, what happens to that model? We're going to measure the fairness and see whether explaining a model causes any differences between the average and worst case fidelity. For example, if you have, male and female patients, and then you see that there's a gap between the fidelity. And if you have multiple groups, so we're not just going to look at sex, maybe we're going to look at self-reported race as well. We're going to look at the average absolute difference in fidelity across all the subgroups. And we're not just going to look at this in the health setting, like, we've been focusing on, right, looking at hospital data, predicting mortality. We're going to do that and look at differences for patients of different sex. We're also going to look at the income prediction data set, the adult income prediction data set, which is a proxy for loan guaranteeing or granting by some banks. We're going to look at, in the education setting, predicting whether a student passes a bar exam. We're going to look at self-reported race as the variable that changes there. And we're also going to look at recidivism risk prediction, which is currently used right now by some commercial software by judges who are trying to make bail decision grants. And so we find that using local and global explanation methods induces subgroup fidelity gaps. So that means when you explain a model, we find that it makes it less fair. Intuitively, this sort of makes sense. If you are telling me that you can snake a decision boundary around all this data in such a way that is as fair as possible, and now you're forcing me to approximate it in some way, it's going to make some small errors maybe. But in this case, we find that it tends to make more errors in the minority classes. And as you explain the model more, the gap increases. And this is not something that you can, fix with data rebalancing, for example. And that we found that this is true because you can detect group information often from the representation space. And so if you try to remove group information from the representation with partial attribute to correlation, it fixes the gap a little bit, but not completely. So hopefully I've convinced you of two things now. Number one, there's amazing things that you could do with machine learning, and lots of people have shown that in health. But also we have to be very careful about exactly how we do this because naive applications would be very problematic. What are safe ways to intervene? Here's one example. If you really just need a simple model, maybe you should make it from the get-go. So if you need something like a decision support checklist because it's easy to use, deploy, and verify, but you don't want to assemble a human panel of experts because it takes a long time, they have to get to consensus, and then years after consensus, a nice medical paper like this will come out that says that consensus that you reached about this checklist or risk score, that's actually going to over and undercorrect for Black patient race every single time. And so why don't we just try to learn optimal predictive checklists if that's what we want in the end? So we propose this in a paper. We form checklist creation as an integer programming problem that directly minimizes errors, but with fairness constraints. Why do I care about fairness constraints? Because if you're trying to predict something like mortality after an ICU treatment like renal replacement therapy, you also want to be fair because you don't want to, like we said before, just say this person won't benefit from this treatment only for female patients, for example, and then systemically deny them treatment. So if we build a model for whether somebody should get continuous renal replacement therapy, right, whether they're likely to die right after it, and so that would help us make a decision about whether they should get it or not, without fairness constraints, we get this model. And I want you to notice here, there's no obvious proxies. It's not using height or weight, which are sometimes proxies for a patient's sex. They are not using creatinine, which is sometimes a proxy for patient's self-reported race. They're not using any of these things that seem obvious. This looks fine. But this checklist actually has a max false positive rate gap between white men and Black women of over 50%. And when you add in fairness constraints, then you can reduce that gap. But you also lose some average overall performance. And these are important constraints that you need to think about in deployed settings like healthcare. We also have been working on how to best repair deployed models when, for example, information changes or EHRs change and suddenly model knowledge becomes stale, rather than trying to reframe from scratch, which can be very expensive and infeasible for a lot of hospitals. We try rerouting knowledge through cached segments of a model's latent space. So if the answer is now different, let's just fix the model's understanding of the answer so that this question and all similar questions will now reroute to the new correct answer. And whereas past work tends to perform really poorly on just tens of edits, it tends to catastrophically forget prior information. We find that our method, GRACE, is able to maintain correct knowledge, both in the thing you're fixing and in things you aren't trying to fix, over thousands of edits, which is really exciting. And we also are working on how we even understand what methods are going to perform well when data inevitably does shift in healthcare. And there's many different kinds of shifts. There could be a spurious correlation where a class and an attribute are tightly linked. There could be attribute imbalance. You only see one kind of attribute across all classes in general. Class imbalance, you tend to see a certain class across all attributes. And attribute generalization, where you just don't observe specific pairs. And examples of these specific kinds of data set shifts are present in many of the state-of-the-art, well-known benchmarks in machine learning settings. And so we benchmarked the best existing algorithms that are out there for doing subgroup shift and group distributionally robust optimization. And we find that existing algorithms actually do a really good job in improving spurious correlations and class imbalance, but they do not at all improve anything for attribute imbalance or attribute generalization. And these are state-of-the-art methods that people are saying, you know, in established work really well. But in general, these kinds of data set shift tend to be, combined within different data settings. And so knowing that, for example, you can perform well given the knowledge we have in these two settings, but not these two, one of the things we try to look at is it the representation or is it the classifier? How is this going to affect our subgroup shift? So we find that just improving the classifier, that's already enough to get better performance in these two cases, the spurious correlations and the class imbalance. And if we improve the representation, it does address a little bit some of the performance that we need to see change in attribute imbalance, but nothing works well for attribute generalization. So this is an open area for future work. And, we really have to think about our metrics in this setting, because we're finding that in most of these papers, everybody's optimizing for worst group accuracy. But worst group accuracy is tightly correlated with things like adjusted accuracy and inversely proportional to worst precision. And in some cases, like our triage example, maybe precision matters more. The final thing I want to cover quickly is let's pretend that we have the best possible model that we can. Let's also pretend that we've made it as robust, private, and fair as possible. It's still going to be wrong sometimes. And let me tell you, it is going to be more wrong on minorities, because that's in general how the data sets we have are, both in terms of imbalance and in terms of practice with societal bias. What if we had a setting like a call summary setting, right? Where you're doing mental health triage and you make one decision. You need to decide whether you should send emergency medical help to a caller's location or call the police. And we've trained people. We said this is a medical response no matter what, unless, if and only if, there's a risk of violence. If there's a risk of violence, you should call the police, and you should only call the police if there's a risk of violence. We surveyed a bunch of people, clinicians and non-clinicians, gave them these instructions. We varied whether we said the person was African-American or Caucasian, and then Muslim or not Muslim. And it turns out, be happy for humanity, regardless of our participants' age, sex, self-reported beliefs about policing, profession, political beliefs, they tended not to call the police disproportionately more often on Black and Muslim people. That's great. And then we trained a GPT model to be biased and give evil advice that you should always call the police on Black and Hispanic people. Black and Muslim people. And so given that we have this GPT model that is giving this evil advice, we also have a control setting where we have a non-evil GPT model that's also giving participants advice. And what I thought was going to happen in this study is that we would find that this evil model disproportionately convinced doctors and non-doctors, maybe doctors less, to follow its advice. And so you got more people calling the police on Black and Muslim people when the evil model gave it advice. And then training the fair model is going to save us, right? That's what I thought. So we have two ways we can give this advice to people. We can give them the if condition. Our AI system has flagged this call for risk of violence, or we could give them the then condition. In this situation, our model thinks you should call for police help. I want to stress to you that these are the same label because it's if and only if. It does not change the machine learning model optimization. It doesn't change anything about the system. It's a tiny deployment detail that honestly, most machine learning model trainers, developers, practitioners would not think about. We would throw this model over to the people who are deploying it and say, you decide exactly how to give this advice to your users. And so what we found is that, as I said, hooray, good day. Respondents are not more likely to call the police on Black and Muslim subjects at baseline. And this is both clinicians and non-clinicians. When you give the biased prescriptive advice to both clinicians and non-clinicians, they are much more likely to listen and call the police. But when you give the same evil advice to people, they retain their original fair decision-making. So this is really shocking for me, and social scientists have really good, potential explanations for this. And this is an effect that you see in some other decision-making settings, in robotics and aviation. Essentially what's happening is when you tell somebody what to do, it's shortcutting their critical thinking skills. And so clinicians and non-clinicians who have these fair decision-making practices, when you give them bad advice, biased advice in an informational way, they retain their critical thinking skills and say, it doesn't track. This doesn't look like there's a risk of violence. But when you tell them what to do, they follow it. This is really important because this means there's no simple fixes for ethical AI in health, right? It's an ongoing process that's going to require really diverse data and teams thinking about a holistic process. So when we're thinking about problem selection or data collection, we need to consider sources of bias in the data. When we think about the way we define outcomes or develop algorithms, we need to think about evaluating these choices and models comprehensively. And when we think about post-deployment considerations, we need to understand not all gaps can be corrected, but models don't have to be perfect in order to be useful, especially if a careful way of deploying the advice allows clinicians to make decisions in a way that isn't affected by biased suggestions. In general, I hope the takeaway that you've gotten from this talk is there's no simple fix. You can't just explain something or fix something, easily. But if we have transparent systems and processes, then we can work towards a set of machine learning methods that can be applied to health. And this is work from a very fantastic group, clinical and technical collaborators who are all supporting this overall vision of creating actionable insights in human health. Thank you. All right, I see a question in the chat. So how do you define algorithmic fairness in settings where the most appropriate treatment should depend on a sensitive category? So this is a really great question. There are some settings where a sensitive attribute or a demographic factor is very biologically meaningful, right? And so, for example, an easy one you can imagine is your gender has a huge implication for whether you have certain kinds of cancers, breast cancer or prostate cancer. And so for those settings, I think algorithmic fairness can be defined in terms of the true rates of the incidence within your own demographic group, because there's a biologically plausible link between this sensitive attribute and the condition itself. There have been many assumptions over the years in medicine that these categories are actually linked to conditions that they're not, right? And so there was a belief that in general, many cardiac conditions were really limited to or tended to exhibit predominantly in men. And that led to a disproportionate loss of female patients when they experienced heart attack. It's not that women don't have heart attacks. It's that clinician, specifically the National Academy of Sciences paper, showed that it's male doctors not recognizing female patients having heart attacks that is killing female patients when they have a heart attack. It's the same thing with, for example, all of these risk scores in the hidden in plain sight paper, where the clinicians posited that there was a link between a patient's self-reported race, for example, and a specific outcome. And so they included it in the risk score, and it turns out it is now leading to over and under corrected risk in all of these different cases. And there's dozens that they point out in this medical paper, and that's actually harming patients. And so I think in some cases, we know there's a link, it has been established, and we should be using it in those settings and think about the right way to incorporate it. In many settings, we have made assumptions, and those assumptions are.
Hey, thanks so much for your talk. And I just want to, maybe my question is a little bit on technicality, but this example with Cybert was so striking. The white person goes to hospital, black person to prison.
Yeah.
It's just like mind blowing. And I'm working on some large NLP models right now. So I was wondering if that Cybert is actually it's not from the pre-trained bird which could be trained on like all sorts of bias data sets. It's Cybert that's like from scratch only.
I think Cybert has been trained from scratch on Common Crawl, if I'm remembering correctly.
I think there is different versions of Cybert. I just wanted to ask. There is Cybert that is from base bird, which obviously could have all sorts of biases. Yeah.
I think we used, if I'm remembering correctly for this paper, the Cybert that is trained on Common Crawl exclusively, not the fine-tuned one. I think in either case, I am still surprised. So I do believe we also benchmarked, there was another BERT model that came out at a very similar time, clinical BERT, which I think is trained on the ICU notes, the hospital and ICU notes. And I think we found other biases within that BERT model, but not the same level of biases. And so I think the source data is very important. But as you have probably seen from the GPT models and the technical report from the OpenAI team, these are inversely proportional, sort of attributes, right? If you don't use enough data, you are probably not going to have good performance on any task you try. But if you use all the data, then you're using data from many sources, some of which are going to carry inherent biases. And so I think no matter what setting you're training your large language model in, I would strongly encourage you to, I don't think anybody expects these models to be perfect. I certainly don't. I am concerned that we are so excited about being more efficient that it feels like, especially with note completion, which I've seen way too many startups proposing right now using some of the GPT models. I don't think that we have that's such a striking and obvious example, but I've seen much more subtle, but probably just as dangerous long-term suggestions from others who have been auditing these models, where the prioritization of different kinds of care is different for people of different genders. The suggestion of what kinds of treatments are appropriate is different for people of different self-reported races. And so I think in all of these settings, we need to realize that medicine is not always practiced equally. And so whether you're looking at medical notes, scientific papers, or, the trash of the internet, there's probably going to be something in there. And you need to just be aware of it, right? It needs to be well understood.
We do have 2 questions in the Q&A. I'll start with the one on the bottom. Do you think the biases will correct themselves over time, or is there a positive feedback loop that will make it worse over time?
I think if we deploy them now, it's a positive feedback loop. I'm being completely honest with you. I think you can correct these things over time with a combination of looking at the data sources we have, understanding how to not naively propagate some of these biases within the training processes. And then also doing some of these very smart HCI considerations studies where you say, okay, we know the model will have some biases. We know that there needs to be some sort of guardrails up around recommendations where it can or cannot make, specific suggestions, or maybe some sort of downstream classifier that's trying to understand when it's giving inappropriate advice. But I'm not seeing that right now. I mean, most recently there was a complaint to the FTC about GPT-4, right, by an organization saying that it shouldn't be used in a lot of these commercial applications where we demand that there is some amount of fairness, right? And so there are rules. For example, there's like federal laws where you have to give loans fairly, right? We have all of these Jim Crow era reform laws that say you have to provide education equally, right? We don't actually have the same kind of like agencies that are out there regulating the performance of medicine equally. We have, ideals and rules within professional bodies and training organizations like the American Medical Association and all these other entities that, have this as a target. But it's not like we have the equivalent of the FTC that goes out there and audits doctors every year and says, this doctor happened to kill every Black patient they saw. There's no list for that. We don't even do what we do with the FAA, which is like actually look at every pilot's flight times and then see the amount of simulation hours that they did and like ask the airline agencies to give us those numbers. And so I would say it's maybe more concerning to me in healthcare than even in financial applications or in education, because we don't have the same laws and active regulatory bodies that are well integrated with practice.
Thank you very much. There is one more question in the Q&A, but we do need to move on to our next speaker. So I think you should be able to answer that. Thank you very much.
All right. Thank you. Bye.
Experimentally validate computationally inferred data from single cell. And our answer to this was through multiplex RNA in situ hybridization stage. Basically, we're going to stain the tissue blocks from the patients we analyzed by single cell RNA sequencing and see if we can find increased frequency of interferon gamma and CXCL 1011 in these tissues. And so we built these. Then what we see is indeed the microsatellite unstable, so these hot MSI tumors had a higher frequency of CXCL 1011 cells, which you see in yellow here. And then when you zoom in on these areas, what's very captivating is that there are these clusters of cells, right? It's not like diffusely scattered, but rather it's these foci of these yellow cells. And in them is where you find enrichment for interferon gamma cells, which you can see in green, and CXCL13 positive T cells, which you see in magenta. And these cells have been associated with tumor specificity for the T cells. So then what you really have is like this multicellular network of cells, and we call this multicellular network the immunity hub. And so then, does this immunity hub matter, is the question. So we find in other cancer types, does it correlate with response? So then we had built this panel, and so then all you really need to do is then find a cohort of patients pre, in this case, PD-1 immunotherapy, and see where you find the hubs. And so we were looking in lung cancer in this new cohort, and what we see is something very similar to what we were seeing in the colon MSI samples, which is the presence of these like foci of CXCL 1011 positive cells, which you see in yellow. So those are like the chemokine expressing cells. And then enriched within these foci are these interferon gamma positive cells interacting with them. So how does this relate to response? So then our question became like, well, how do we think about multicellular networks? How do we think about spatial aggregates in tissue? So our favorite approach to this is to essentially like lay a grid over your tissue and then kind of go window by window, seeing whether or not there is CXCL 1011 in any given window. And so like the way we did this under the hood was k-means clustering k = 2 solution. And then the question is then like, well, these are individual windows. Can we then aggregate the windows that are touching to construct contours of these immunity hubs? And so we do that. And then when you look at the percentage of tumor area covered by these immunity hubs, you see that responders have a higher frequency than the non-responders. So like, I'm an experimental biologist, if you can't tell by now. So I wanted to know, like, well, what's in this immunity hub? Like, what's kind of the biology underlying it? And I'm very interested in CD8 T cells. So made another panel with CD8 T cells. And to simplify, like CD8 T cell developmental trajectories in the setting of cancer or chronic antigen challenge, there's a particular differentiation trajectory. And so I'm going to outline that for you. It starts with the naive T cell, which expresses a transcription factor called TCF7. It does not express a co-inhibitory molecule called PD-1, which is obviously target of PD-1 blockade. And then upon activation, cells transiently express the TCF7 and PD-1 together. And then over time, PD-1 is increased and TCF7 is decreased. And then you lose effector molecules, there's cytokines known as IL-2, TNF-alpha, and interferon gamma. And then these cells become less and less effective. And so sometimes people like to call these cells exhausted. And these cells have their chromatin remodeled. And like, why is that important? That's important because like PD-1 blockade immunotherapy, it cannot take these cells that are exhausted and make them young again. They can't swim back up the waterfall. What happens is that these cells that are more plastic, that are higher upstream, they serve sort of as like the wellspring of anti-tumor cells that expand and invigorate the anti-tumor immune response. So then what we do is we have our two panels, computationally co-register them, and then we were able to do this for around 2/3 of our cases. We then go back to this like laying the grid approach, and then we have taken a page essentially from the single cell RNA-seq community, and we perform clustering. And the way that we do this is somewhat unusual in that each dot here represents an immunity hub, and then we look at the like phenotypic composition of each hub. And so what are these clusters telling us? Well, one of the interesting things is even though we didn't feed responder and non-responder information into these phenotypic clustering, what we see are differences between the responders and non-responders. So, and you can't really see my mouse, but if you look at the non-responders, you see that especially sort of from the middle of the non-responders over to the right, there's a much lower frequency of immunity hubs. That is that on the y-axis, see essentially the frequency of immunity hubs, and then the responders, you see that it's higher. But what's like catches the eye about the responders is actually that there's more of these like pink and orange bars. And so like, what are these pink and orange bars? If you look over in A, they essentially correlate with this cluster 3 and this cluster 1. And what's interesting is you can start looking at like which clusters are enriched in responders, and what you see 3 is like most enriched. And then if you look in D, you can see the fraction of tumor area covered by subcluster 3 is much higher, actually, with the exception of one blue circle up in the left. And so then I was kind of curious about this particular patient. And so I asked the thoracic oncologist, if they knew anything about this patient. It turns out this patient was, according to RECIST criteria, a non-responder because they had a brain met that then subsequently received radiation. But like actually the thoracic oncologist had been sort of like touring the lecture circuit, touting like how well this patient had responded to therapy. And so it kind of became a very interesting case that this patient had lung lesion, which you can see with the red arrow at upper left. And then you also on the left side, nodal disease. And the patient received only a single dose of PD-1 blockade therapy because they developed an adverse reaction with pneumonitis. But even after one single dose and no other systemic anti-tumor therapy, they're still alive 5.5 years later, and don't show signs of progression of their disease. And so that is kind of reflected when we look at the survival of these patients. So what you see in E and F is E is like progression-free survival, and F is the overall survival of these patients if you have even 0.1% of your tumor area covered by this subcluster 3. And so then again, I'm a biologist, so I'm like, what is in this subcluster 3? Like, what's the special sauce? And so what's interesting is that subcluster 3 was enriched for these sort of like wellspring cells, which were the cells towards the top of the waterfall plot that I showed you earlier. And then they were also enriched along with the orange subcluster for interferon gamma expression. So I'm kind of like well, I'm a pathologist and I'm like an imaging convert. So one of the nice things about imaging, as opposed to the single cell data, is you can like drive back to the spot on the slide that precipitated a given call, and you say, well, what does it look like? And so what you can see like in these white ovals are the yellow CXCL 1011 cells. That's what you need in our system to be called an immunity hub. And then you also see enrichment for these cyan positive CD8 T cells. And I don't. So like the TCF7 again is a transcription factor, so expressed in the nucleus of cells. And so that's stained in red here. And you can see that many of the CD8 cells are TCF7 positive, which goes along with what we saw in our clustering. And then, so I don't know how many people here are like aficionados in spatial imaging, but when you see a lot of like aggregates of immune cells in a tumor, you think of this structure that's called a tertiary lymphoid structure, which is kind of a little bit like you're making a lymph node inside of the tumor. But what you see when you have like the mature version of tertiary lymphoid structures, you see like these like B cell follicles or germinal centers, which are like where antibody immune response is made by B cells. And so like I, being a pathologist, stained for an H&E and looked and saw like, we don't really see germinal centers or B cell follicles here. And so to pursue this more, we build a third panel and stain a subset of cases with a panel that includes MS4A1, which marks CD20, which is like a B cell marker. And sort of bottom line is when you look at the cyan here, you see there are some B cells present, but there aren't these like big aggregates of them into germinal cell structures. Additionally, thinking about how tissue is organized, again, around these ideas of like chemotactic cytokines pulling the cells into their places. CD8 TCF7 cells have been shown to migrate in response to CCL19, so we also included CCL19. And we saw in these areas of the subcluster 3, you see a lot of CCL19 expression. Additionally, side note, some of the cells that are most well known to express CCL19 are activated dendritic cells, which can also be marked by CCL22. We include CCL22 as a marker, and we see that there might be these activated dendritic cells present, which I'll kind of circle back to in a sec. But anyway, like the point is that in these areas, this subcluster 3, which is very favorable, we see the core immunity hub, and we also see these stem-like CD8 TCF7 cells. So being very, very creative, we call this the stem immunity hub. And this area is also enriched for CCL19. So we kind of have a picture of how the tissue might be organized. And And so, actually, I'll skip this just in the interest of time. But basically, we did some staining with a platform called Geomics that allowed us to see the difference between the stem immunity hubs and tertiary lymphoid structures. And what we find is that tertiary lymphoid structures are essentially enriched for B cell genes, and that stem immunity hubs are enriched for, of course, CXCL10, one of the things that you need to be called an immunity hub, and also the closely related chemotactic cytokine CXCL9 and interferon stimulated genes. And so now I want to get to the part that I want to talk about. So we can start to look at like how cells are talking to each other with a newer technology that is called MERFISH, which allows you to look at the expression of 500 genes simultaneously, and allows you to look at it at single cell resolution. So then we took a small subset of 4 cases that have these stem immunity hubs, and then we can do 2 kinds of analysis that are not mutually exclusive. The first is to use this window or tiling based approach, and the second is just to look at cell by cell and do like a clustering based approach like you would with single cell RNA-seq, and then you can like overlay and see who's enriched where. And when you do that, you cluster the tiles and you see that there's one cluster here, 7, that is enriched in CXCL 9, 10, 11, and in CCL 19 on the y-axis. So if you take an unbiased approach, you can still recover back these stem immunity hubs. And as I described earlier, that we see a similar set of genes. So we see markers of stemness like CCR7, CD62L, IL-7 receptor, and TCF7. And then we see some markers for like B cells. So basically, using this unbiased approach, we kind of recover what we had found in our initial staining and also in our geomics data, which I touched upon briefly. So then we can start looking at the spatial layout of these areas. And like the core question that we want to get at is like, well, what's enriched in these stem immunity hubs and who's talking to whom? And so what you can see at left is this plot of enrichment. So if you look at the x-axis, you see like things that are enriched in the stem immunity hub versus the rest of the tumor, and the y-axis is things enriched in the immunity hub that's the not stem immunity hub versus the rest of the tumor. And if you look at the right side in blue, I have the cell types that are enriched in this stem immunity hub, and sort of reassuringly, we see these stem-like CD8 TCF7 cells are enriched. We also see enrichment of CD4 T cells and regulatory T cells. And then the thing I kind of want to call your attention to that I boxed in red are these activated dendritic cells. So I don't know who here has had immunology or not immunology, but basically activated dendritic cells, they're important for activating T cells. And so dendritic cells are professional antigen presentation cells. And so they will interact with T cells and can activate these cells. So it's very interesting that they would be enriched in this particular stem immunity hub. And additionally, their gene expression profile is very well known from single cell studies. And this includes what I've boxed at the right here, CCL19, CCL17, 22, which are all T cell attracting chemokines, and also IL-15, which is a cytokine that can support memory and stem-like T cells. So then we're able to start sort of putting the biology together of how the cells are enriched in the stem immunity hub and what factors they might produce. And then lastly, the interesting thing when you have like single cell resolution transcriptomic data is that you can start to think about which cells interact with which other cells. So in this schematic, which we took from Michelangelo's group, you can imagine you're trying to figure out how often is phenotype 1, the green cell, talking to phenotype 2, the red cell. And you can you can count the frequency observed, but you can also hold the green cell's position constant and then scramble the identities of all the neighboring cells to get a randomized distribution. And then you can see in that like histogram, you can compare your actual observed distribution versus your randomized distribution, see if you're seeing more frequent interactions than you would expect by chance. So when we do this, what we see here is a plot where the stars indicate significant interactions, and the x-axis, or the rows, are the index cells. And so you can look across, for example, at the second row from the top, the activated dendritic cells, and you see that they have a higher, they're enriched for interactions with CD4 cells and Tregs, and they're not enriched for interactions with CD8 T cells. So we kind of know which T cells these dendritic cells like to interact with. In contrast, from this single cell data, we also find who's expressing the CXCL 9, 10, 11. Turns out that it's macrophages. You can see CXCL 10 positive macrophages at the bottom row, and you can see that they don't actually like to interact that much with CD4 cells, but rather they preferentially interact with CD8 T cells. So then by integrating sort of the data we've taken across these different imaging modalities, we can construct what is in a stem immunity hub and what are their interactions. Again, at the center of this is the immunity hub, which is the CXCL 1011 interferon gamma circuit. We know now that macrophages produce the CXCL 9, 10, 11 and interact preferentially with CD8s. We know that the stem immunity hub is enriched for stem-like T cells, and also for these activated antigen-presenting cells that preferentially interact with regulatory T cells and CD4 T cells. So actually, I just summarized that for you. So basically, I guess I'm at time, so I want to thank you for your attention. I don't know if I have my colleagues and collaborators here, but I'd like to particularly thank Karen Pelka, Matan Hoffrey, Linda Neiman, and Max Spurl for this great productive teamwork, and then also Nir Cohen for mentorship. So thanks. I'm happy to take questions.
Thanks, Sean. That was great. I had a question. So enriched in your hubs were TCF7 PD1 positive.
Yeah.
And that's the marker, the stem marker,
Right.
and the exhaustion marker.
Oh, the waterfall.
Yes, the waterfall.
The waterfall. Right. Yeah. So basically, at the top of the waterfall are the most plastic cells. It's kind of like I don't know, any developmental trajectory in a way. Like when you start out, you're young and plastic, and as time goes by, you become more locked in your ways.
So the reason PD1 blockade works is because you then push them down the other.
The reason, yeah, so these cells that are towards the top of the waterfall, okay, some proportion of these are antigen specific or tumor specific. And the way that PD1 blockade works is that these cells then preferentially expand, then go and fight the tumor. So that's a series of papers over the past, I don't know, 4 years, 5 years now in mouse and human. So a lot of work with adaptive transfer models. You sort the cells from different states, and then you transfer them, and you see which one leads to better response. And they've done this now also with Steve Rosenberg at NIH with humans, has found a similar pattern. And then obviously in mice, you can also label cells so you can track them, and you can also knock out these genes. So it's approaching canonical at this point.
Thanks.
Thank you for your lecture. I have a question about do you think we can find biomarkers that can give some suggestion to patients, such as some patients come to us, we can give some suggestion to them, like what kind of immunotherapy is truly beneficial to their cancer type or?
Yeah, yes. Thank you for asking that question, because I guess it's a way that we can tie into the theme of this, meeting. So yeah, how can you build then a biomarker that then you could sort of train to help you figure out what should a patient receive? So there's a few things that are implicit in this, right? Because you're trying to figure out what's the biology underlying the response, and then what are the favorable features that go with the response. And of course, I was kind of spent a substantial portion of the talk today talking about these CXCL 1011 positive cells, right? Because I think that they're something that people see in bulk RNA-seq data. So like if you took like blocks from, tissue blocks from patients who went on to receive therapy, and you cut them and you like looked for gene expression, you could do that in the absence of like spatial information. And these chemokines, CXCL 9, 10, 11, and also these these interferon stimulated genes actually in this plot here that I touched, here that I touched upon. These genes in blue are many of the genes that whose expression is associated in non-spatial data with response to PD-1 blockade. And now we understand basically the expression of these genes is organized spatially into these clusters, right? And so that allows us to think about like, well, you can look at this without spatial information. If you have spatial information, can you see like the immunity hub and build markers off of that? And I think the answer is, yeah, you could do that. And it might be, it's more of like a rational biomarker design than you would have just by like staining with whatever antibodies you can kind of have on hand. And so like, we've done some work with like CXCL, 10, 11 and interferon gamma. And when you do just like that kind of staining, like the first RNA-ish panel, you can outperform like a PD-L1 IHC, which is kind of like what people, what we use clinically.
Thank you very much, Dr. Chen.
Thanks.
Our next speaker is Jayashree Kalpathy-Cramer.
So thank you so much for the invitation to be here today. I was at MGH till last year. I've recently moved to the University of Colorado, and the work that we've been doing over the last decade or more has been in the applications of machine learning to medical imaging. We first did a lot of work in radiology and oncology, and now we're doing a lot of work in ophthalmology. And I guess the one thing I would say is that the problems are very similar, and no matter what our domain is, we run up against almost the same kind of things, no matter what we work with. And so from a technical perspective, it's great because we solve the problem once for one domain and write a paper and then apply it to a different domain and write the same paper again. So the first disease I'm going to talk about that we've been working a lot, again, more than a decade now, is something called retinopathy of prematurity. It is one of the leading causes of preventable childhood blindness worldwide, both in the US and worldwide. The good thing about this disease is there's a lot of really good options from a treatment perspective if it's diagnosed early. And so one of the challenges, both again in the US and worldwide, is that there's not enough pediatric ophthalmologists who are willing to diagnose and treat this disease. So the question is there something we can do from a machine learning perspective to train an algorithm to help with this process and sort of standardize the diagnostic process? So we've been NIH-funded consortium that's been working in this area for more than a decade now with people across the globe. This disease, one of the sort of fundamental causes is sort of improper oxygen in the NICUs. So fortunately, a lot of the premature babies are surviving much, much earlier than they used to. Unfortunately, sometimes, especially in places that don't have like really good equipment in terms of monitoring the oxygen delivery, too much oxygen can cause these blood vessels to proliferate in the eye and cause ROP. And so one of the things that, again, we are hoping to do is if we can do sort of timely and accurate ROP screening, and that might allow us to catch the disease and treat it and prevent these babies from going blind. The one reason sort of motivating this, one reason we would like to do that is the diagnosis of ROP is quite subjective. And this is not unique to ROP. We've seen this for sort of many different diseases, is that if you take the same image and ask a clinician to read it, you might get slightly different answers. We don't like to necessarily acknowledge it, but that's often our experience. So we've done the study where we took the same image, asked 8 clinicians across, 8 experts across the country to grade them, found sort of a wide range of responses. And we did that in ROP, we did that in COVID, we've done that in pretty much many, many diseases, and we see sort of a lot of variability. There are some people in the audience who've studied this problem extensively. Another problem is that the exams are quite stressful to the baby. So these are tiny, tiny infants. They're extremely low birth weight. They have all kinds of health issues. And now we want the clinician to go put this the lens in the eye and sort of look, and it can be quite stressful. So if we can be quite sure that this baby is not at risk for developing ROP, we can at least greatly reduce the frequency of these exams. So one of the things, again, we'd like to do is have a much better predictor of which of these babies is most at risk for developing ROP. This is a map of the number of ophthalmologists per population, and you can see that in many parts of the globe, there really aren't enough trained ophthalmologists to treat this condition. So again, if we can sort of identify those babies most at risk, we can then divert the resources accordingly. And then finally is the sort of understanding of biology and the outcomes. So one thing we noticed again in the study where we looked at a severity score for the disease to see when people treat, what we find is different clinicians treat at very different points. So some people are much more sort of aggressive about treating early, and others have a much more wait and watch kind of approach. And this allowing AI to sort of quantify disease severity objectively can allow us to explore these sort of differences we see between clinicians. The way this disease is diagnosed is by looking at the pictures of the back of the eye. You can look at the vasculature, and as the disease progresses, the vessels go from being sort of straight to being tortuous and dilated. And it's a very relatively straightforward task from an image analysis perspective. It's essentially you're looking at things like dilation and tortuosity. And more than 5 years ago now, we trained a deep learning algorithm for classification. We had a bunch of experts from across the globe do annotations on this. And then we trained a fairly straightforward classifier and were able to get very high diagnostic accuracy. One thing you will note is if you plot each clinician's performance on the ROC curve, they lie different parts of the ROC curve. So some people are much more sensitive, some people are much more specific, and that you can see when you give them the same set of images and ask them to grade them as well. We also looked at the inter-rater performance that you can see in the chart on the right, and it's can be quite variable. And at least our algorithm is well in the middle of that performance. This algorithm went through the FDA got the breakthrough process, and it's in the process of being commercialized. And again, we were very excited and happy. So that's step one, everything looked great. The second problem that we are working on, I'm going to switch domains completely, is cervical cancer screening. This is a problem we are working again with the National Cancer Institute. Currently, we're in a large moonshot funded project, and the goal is to screen more than 100,000 women across the globe over the next 2 years. So cervical cancer is a leading cause of cancer morbidity and mortality. It's really a problem in places with HPV infections where the vaccine hasn't penetrated, in terms of uptake hasn't been not quite like the US. And again, screening can help. It's great because if you screen and identify these patients, you can treat and greatly resolve side effects from that. The plan with this program is to have a sort of a two-step stage. There's a self-sampled HPV typing as step one, and then for those women who are HPV positive, we then have a is known as automated visual examination, where acetic acid is applied to the cervix, and essentially an algorithm will try to analyze the images to say if this patient is likely high risk or low risk. And right on the spot, the patients with high risk potentially will be treated. Large consortium across the globe that working on this problem. The early work was done at NCI and training an algorithm for the AVE, and it showed extremely high diagnostic accuracy. So again, we had something that looked great. Lots of papers were published, everything looked wonderful. So as we keep talking about, and it's getting more and easier every day, it's getting really easy to train these algorithms, right? You have ChatGPT 4, it'll write your code for you. Your auto ChatGPT will improve it, get it better over time. It on the right side is a abstract it spat out for me for my publication. It looked really good. It looked a lot like ones we published. I suspect it was trained on some of our publications. And it looks really easy to do that. But to find these algorithms that are sort of broad and general, robust, unbiased, we heard about the questions of fairness and bias earlier today, self-aware, it still continues to be quite challenging. So many of these algorithms don't have a good sense of their self-doubt. They'll happily be wrong, very confidently wrong, and they're not very calibrated very well to say, I shouldn't answer here. So some of the challenges, so going to some of the practical challenges when we went to real world deployment, for the ROP, for instance, when we had, so different populations, if you look at the back of the eye, the pigmentation can be different. So if you train your algorithm in the data set that was trained of patients in the US and now mostly white population, and then you go to places like India or China, the fundus looks quite different and the algorithm may not work as well. The different cameras look quite different. So the field of view can be different. Just the image qualities can be quite different. Protocols are different. So we were expecting to see fundus photographs in our data set, and suddenly we saw something that is not a fundus photo, but it looked a lot like a fundus photo. So when we ran the algorithm, it happily gave us an answer. Image quality can be an issue. The biology can be different in different parts of the world. Almost very similar problems were then subsequently noted in the cervical cancer world as well. And a subsequent publication came out actually highlighting many of the challenges when you go from having what looked like a great algorithm on the first data set, and then you try to apply it sort of worldwide in very different populations and different domains. So we've sort of come up with a list of 7 things that we worry about when we translate our algorithms. So the generalizability question, are these algorithms brittle? When you train them on one population and then you try to apply them to a different population, whether it's because of different prevalence, different scanner types, different anything, does the algorithm still continue to work? One surprising thing that, again, Katie worked on was the notion of repeatability. One thing that we've been surprised to see is that actually not very much literature that says how repeatable these decisions are. So if you had the same person, you image them twice with some slight differences, different positions, slight variations, do the algorithms give you the same answer? And surprisingly, the answer is not often. So if nothing else, people should at least study the retest performance of these algorithms. The notion of a gray zone. Many diseases are not binary. There's a nice intermediate zone, but we often don't appreciate the fact that diseases lie on a spectrum, and there's this great tendency to dichotomize everything. And that causes us to lose a lot of information in the middle. Calibration and silent failures, explainability, we heard about that in the first, again, sort of overfitting. This is very common in the ML literature, and the questions of bias, and especially in ways that are hard for humans to appreciate, was again covered beautifully in the morning. So I'll focus on the first 3 of those. So the brittleness has been reported in the medical imaging literature a little bit, and we're starting to see a little more of that in terms of looking at external validation. But very often the studies that are published in the literature tend to be validated sort of on the very same population that they were trained on. And that sort of many different forms of data heterogeneity, concept shifts and other drifts can occur. So you might have very different prevalences of disease, and that might be because of true differences in prevalence, or it could be because the annotators are different. And when we've done these sort of studies where we look at prevalence of something across institutions, where we expect the same distribution of disease classes, we find that's not the case. Some people don't use an intermediate class, some people use more intermediate classes. So people might say mild, moderate, severe, or mild, moderate, extremely moderate, severe kind of thing. So the way people bin these diseases can be quite different across institutions. Scanner types can be different. And even more troublesome is that the association between disease and certain confounding factors can be different in different populations. So all of these things lead to different performance in different subpopulations. Again, the first speaker talked a lot about looking at these sort of differences and evaluating them carefully. In terms of practical, this was work again done by the NCI. They looked at the performance of the cervical algorithm. When they trained and test on the same data set, as you see in the diagonal elements, you get really high performance. But when you train on one data set and test on a different data set, the performance is just completely close to random sometimes. If you happen to have both data sets and train an algorithm, it gets good performance in both. But now if you have a third data set, it may not work as well anymore. So the ability for these algorithms to generalize sort of outside their training data has been somewhat limited. Hopefully that's changing now with some of the more newer algorithms. This is the same image of the same woman in the top row on different cameras, and you can see it looks quite different. In the bottom row, you can see a whole host of cameras. And again, you can appreciate that the imaging characteristics can be quite different. So algorithms trained on one may not generalize very well to the others. This is a similar thing that we saw from the ROP case, where we had data from a North American population as well as a population from Nepal. When you look at things like UMAPs or T-SNEs, you see that the clusters of the features are quite different. So very often these algorithms seem to learn the device first, and within each device, they then learn the pathology. What we had hoped instead is that there'd be more disentanglement of the features earlier on, or it'll first learn to normalize the device-related attributes, but wasn't always the case. And again, you can see that when you train and test on the same population, you get great performance, but you train on one and test on a different population, the performance really drops quite substantially. So different ways in which we're thinking about it, obviously having data from many different scanners, different populations can be really helpful. And there are many efforts nationally to try to create these multi-institutional data sets. Some way of doing out of distribution detection. So you've trained your model, you know what the training data look like, you're going to apply it to a population, making sure that the data you're applying it to looks sufficiently similar to the population that it was trained on might be at least one of the guardrails we might want to consider. Lots of different methods coming, self-supervision GANs might help with some of this generalization issue. We are particularly interested in things like federated learning, where we might be able to train from multi-institutional data sets without the need for data sharing, and hopefully get a much more diverse data set. The second problem I want to touch, completely messed up. I guess Google Slides to PowerPoint does not work, or vice versa. So those two images are the same image, same person, look very similar. In one case, the prediction of the model was cancer. The other case, it was not cancer. If you plotted a test-retest probability of disease, you often see a chart that looks scary like this, in the sense that you expect everything to lie on a diagonal, meaning you take two images of the same person, you should get the same prediction. But what you see is a lot of data points that are off diagonal, which is not good. This was, again, we've done this in 4 domains. So we did this for cancer, we did this for knee x-rays, we did it for ROP, we did it for breast density estimations. And many cases, if you have this sort of, you plot the test, retest, what you really want is a nice tight line around the diagonal. What you often see is this sort of scatter plot that looks pretty scary. One way in which we look to address this is the use of Monte Carlo dropout type of models. So by doing these approaches where you're sort of ensembling many different models, you get much more stable predictions. And this really greatly reduces the variance around for each person's measurement. And this was true again from many different diseases that we found. The same sort of thing. So whether you're looking at a binary model, a multi-class model, a regression model, for all of these different model types, adding the Monte Carlo really seemed to help the tightness of the test-retest performance. The third problem I wanted to touch upon is this notion of continuous severity. A real world is a continuous spectrum. This is a color-coded chart of annotations by 8 different world experts in ROP. So each row is a person, each column is an image. The color indicates the disease severity. So what you'd hope is nice tight bands that are with a boundary that's very clear between the disease classes, but what you really see is a pretty fuzzy boundary between sort of mild, moderate, and severe. And the boundary seems to be quite different for different people. For some people, it's much more to the right of the image. So that like the bottom person has a lot of reds and almost no greens. The top person has quite a few greens, some yellows, and very few reds. So the same image read by 8 people can give you very different answers. The top one was for ROP, the second one was for COVID. And again, they lie on different parts of the ROC curve as we see it repeatedly published in the literature. So one way to think about doing this is framing it instead as a, instead of saying, is it mild, moderate, or severe, ask them, do you like the right one or left one better? And based on that, you get a ranking. And the ranking tends to be much more consistent. So instead of doing this on a class scale, you do it on a severity scale. And once you have a severity scale, you can do all kinds of nice things with it. You can use it for screening. We've done, again, using the screening to say that it works quite well for disease classification. Because you have a nice quantity to severity scale, you can look at things like disease monitoring. So is it getting better? Is it getting worse? That by doing that, you can identify the patients that can be screened much less frequently and actually reduce the amount of screening that's required. So you can have almost perfect sensitivity. And by doing that, you can greatly reduce the number of exams needed. We can also look at things like hospitalization, hospitals across India in this case. So in certain hospitals, we see much more severe disease for the birth weight that we expect. So we can use it for monitoring sort of health systems. And so that's sort of our, we strongly recommend the use of a disease severity scale. From the cervical standpoint, what we found is that we really want good reproducibility, good discrimination. It has to be well calibrated and potentially sort of go across devices. So we have this sort of a funnel of evaluating algorithm performance, looking at these different criteria. And based on that, we have an algorithm that we think works quite well in terms of being repeatable, having good discrimination, being well calibrated, and having a good sense of uncertainty. So as to summarize, I think this is our mental checklist of the things we think about when we go from having something that looks good on our first publication to something that is going to work in the real world for 100,000 women across the globe. Is things like retest repeatability, the reproducibility across devices, the things like calibration, is it confidently wrong? Other ways, does a model look at things like quality? How does it work if the images are not of good quality, for instance? And I guess it's gone off the screen, but yeah, and with that, thanks to everyone in the lab who helped with a lot of this work. So thank you.
Anyone have any questions? I don't see any online.
Hi, thanks so much. Can you go back to the slide where you showed that what do you expected test retest to be like bound? Yeah, you got very scatter plotting. I just wonder if those models were trained with the robustness to retest, like augmentations, all sorts of like ML practitioners stuff, best practices stuff.
Absolutely.
So they were, and still you get the biological retest gets you all over.
Exactly.
Oh,
exactly. So that's exactly the scary part, right? We did all of these usual augmentations, everything that we expect will be working from a machine learning perspective, but when you do it for real, we find that not to be the case.
Yeah, this is striking for sure. Yeah, thank you.
Thanks.
Hi. Do you think learning like the intermediate pathology, like morphological traits and stuff would help with generalizability?
Absolutely. It helps with a lot of things. So instead of doing binary classes for the cervical, we found that going to an intermediate gray zone was extremely important for reducing grave errors. So what we really don't want to do is call something that's normal, something that's pre-cancer normal. And by adding this intermediate zone, it really helped us learn sort of a spectrum of disease. So absolutely, yeah.
Thank you very much. Now I would like to welcome Bill Lauder.
Well, it's great to speak to you all today. So my name is Bill Lauder, and I'm a new faculty member at Dana-Farber and Harvard. And prior to that, I actually came from industry. So I was a co-founder in a startup that worked on AI for mammography. And so I'll be talking about this area of AI for mammography today. Okay, so at a very high level, if we were to consider mammography in an AI framework, it might look something like this. We'd have a mammogram image, which is an X-ray of the breast, and some type of deep neural network that would process this image and ultimately try to predict if breast cancer is present or not. And now the whole crux of deep learning is this notion of end-to-end optimization. If we collect enough training data of mammograms where we know if breast cancer is present or not, we can train the network in an end-to-end fashion, updating essentially each layer in the network to form this end goal of breast cancer prediction. If we zoom out though, this model training step is just one step in the full pipeline of clinical integration. And so I show this image as kind of I like to think of it as a useful metaphor of as we're trying to clinically deploy algorithms, this end-to-end notion that's useful in deep learning training is also a way to think about clinical algorithms as well. So essentially, if we want to optimize for clinical deployment, there are questions that other speakers have raised around robustness and generalizability, questions of UI and interpretability, and even basic questions such as the types of data we ultimately want to be able to process ultimately depend on this end goal. And so today I'll essentially do a deep dive along this pipeline for mammography. But as Jayashree and others have mentioned, the kind of challenges that are in one domain often apply to other domains as well. Okay, so, starting off with the problem definition, why did we start with mammography to begin with? In short, it was because of the high potential for impact. So mammography is a screening modality where women get a screening exam every 1 to 2 years. And it's been shown to significantly reduce breast cancer mortality, but it's estimated that maybe around 10 to 30% of breast cancers are initially missed. And overall, there's limited access to screening worldwide. And so if we look at a full mammogram, such as this one, sorry, I don't have my slides here, so I'm gonna keep looking back and forth, partly going off of memorization. But so if you look at a full mammogram, which is 2 images of each breast, it becomes clear why there's room for improvement. So this is actually an example of a patient with breast cancer. And to my naive eye, at least, nothing immediately jumps out as to the location of the cancer. And so to see the malignant lesion, you really have to zoom in. And so these tiny white specks are called microcalcifications, and they can be a sign of cancer. So overall, interpreting a mammogram is it's a needle in a haystack problem. And this needle in haystack nature is exacerbated in something that's called digital breast tomosynthesis, where instead of just having one 2D image per view, you can have up to 100 images, which are each at a slightly different angle of the breast. And so while this added dimensionality, it adds more data to parse through, it also can be super helpful in detecting subtle lesions. And just as this needle in the haystack nature can be challenging to the human visual system, it's also can be challenging in developing AI algorithms. So you can imagine if we try to train an AI algorithm to say breast cancer is present somewhere in this image, it could easily pick up on features that aren't actually indicative of cancer. So things that kind of pop out but aren't actually lesions. And so this notion of training with kind of just an image level label is known as weakly supervised training. And this is opposed to strongly labeled data where we have annotations of like the present where the lesions are located. And so the strong annotations can certainly help in training your algorithm, but they're also super costly to collect and sometimes infeasible in some domains. So ideally, we'd be able to train with both weakly and strongly labeled data. And just as this kind of localization-based interpretability is important in terms of the AI model training, it's also a useful aspect in thinking through the AI model's outputs. So if the AI model can localize the lesion, you can imagine that that's much more clinically effective than just saying breast cancer somewhere present. Okay, so to summarize, screening mammography certainly saves lives, but there's much room for improvement in terms of interpretation accuracy and access to screening worldwide. And to do so, we need to overcome this needle in a haystack nature while simultaneously maintaining some type of interpretability, at least at a location-based level. And ultimately, we'd love to have algorithms that work for both 2D and 3D mammography. Okay, so now moving on to our approach. So at the time, we collected data from a number of different sources. Some of these data sets were publicly available. Other data sets were collected from clinical sites that we partnered with. And overall, it encompassed both 2D and 3D mammography, where at the time, some of the 2D data was strongly annotated with these bounding box annotations. But all the 3D data was only weakly annotated at the time. And in total, we had around 100,000 exams and 7,000 cancers. So with this data set, moving on to algorithm development. So back before the ChatGPT days, when I started first working on this problem, I tried kind of this brute force approach of taking a network that had been optimized for, say, ImageNet or natural images and trying to directly apply it to mammograms. And as you can imagine about this whole needle haystack nature, this approach just didn't work well and the model kind of simply overfit. And so what I did is I went back to the drawing board and tried to understand the problem better and specifically read a couple breast imaging textbooks. And so when I read these textbooks, I noticed a pattern in kind of how they laid out how to read a mammogram from a kind of human perspective. And so they'd often start by describing different types of lesions, masses, and calcifications, and show examples of what they look like. And then they'd move on to describing how to search an entire mammogram to find these lesions. And then when it got to this 3D mammography, they'd always emphasize the importance of interpreting some type of 2D imaging in addition to the 3D slices. And so this 2D imaging that encompasses a 3D exam could be either a conventional 2D acquisition, completely separate from the 3D acquisition, as illustrated on, I guess, your right. Or most equipment these days also offers an option to generate a so-called synthetic 2D image, shown on the left. And so how the synthetic image is created is actually from the 3D slices themselves. So they essentially take this big stack of 3D slices and then use some image processing techniques to collapse it into just one 2D plane to try to kind of make it look like a 2D image was acquired. Okay, so now moving on to the algorithm. So with these intuitions of mine, I essentially developed an approach that consists of training a core deep learning model in a series of stages, where each of these stages tries to mimic the strategies used by radiologists in learning how to read a mammogram. And so the first stage consists of training a patch level model that takes in a cropped image patch from the mammogram, and it tries to predict whether a lesion is present in this given patch. And so you can imagine that this is kind of learning to, knowing what a lesion looks like before learning to read a full mammogram. And then next, for the second stage, this patch level network is used to initialize the backbone of another network that now reads the full 2D image, and it outputs bounding boxes over predicted lesions. And so in this case, by initializing this network with the patch level network, it's kind of akin to using this network in a convolutional fashion to scan over the entire image to find these lesions. And importantly, for these first two stages, they rely on the strongly labeled data, these bounding box annotations, in order to train these models. And then finally, the third stage makes use of all the data available by training in a weakly supervised fashion, where you just have a binary label if cancer is present or not. And so to do this, we also need to generate essentially an image level score from the model as well. And the approach I took here is relatively straightforward in taking essentially the image level score as the max score over all the bounding boxes in the image for the model. And so while it's essentially a simple approach, it has a number of advantages. Most critically, it maintains the localization-based ability of this detection-based network, and that under the hood, it's still generating bounding boxes, so we can then still go back and see where it localized lesions. So with that, we have a fully trained 2D model. Now moving on to a model for 3D, this DBT. So with only having weakly labeled data at the time, I was particularly inspired by this idea of a synthetic 2D image. So if we could essentially take the 3D problem and collapse it to a 2D problem, that could really help prevent this overfitting. But at the same time, we don't want to just do that naively, because we could lose a lot of useful information in the 3D data. And so I essentially decided to make a more optimized synthetic 2D image by using AI. And so the approach goes like this. From the full stack of 3D slices, we run the 2D model on each slice. And so that's going to generate bounding boxes on certain slices. And then we project the boxes at each kind of XY position down into one single 2D plane. So kind of stitching these patches together in an approach that we call a maximum suspicion projection. Essentially using this AI model to find the most suspicious regions everywhere and condense that into one single plane. And just give a quick example of what this can look like. So here's an example of the MSP on your right compared to a conventional synthetic 2D image on your left. And while it's definitely very subtle, in the MSP, you can see these so-called speculations, which are these kind of like star-like projections radiating from this mass in this case. And these speculations are a telltale sign of cancer. So this is a patient with breast cancer. And compared to the default synthetic, these speculations are much less apparent. And so one reason you may imagine this is the case is that this 2D synthetic by default is trying to create as if an actual 2D acquisition was acquired, which is loosely analogous to kind of averaging across all the 3D slices. So if there was tissue above or below the lesion, it could essentially mask these so-called speculations. Okay, so with this MSP approach that essentially converts the 3D problem into a 2D problem, we can generate these MSPs on the training data set of 3D imaging. And then we can just proceed as if we had a 2D model and fine tune the 2D model on these images themselves. And so with that, we kind of approach that encompasses both 2D and 3D data, while making use of both strongly and weakly labeled data, while critically maintaining this localization-based interpretability and still in terms of still outputting bounding boxes. Okay, so now we have an algorithm, but there's still a number of considerations of translating this algorithm into something that's actually clinically usable. And so we set out to build a product that effectively assists radiologists as they're interpreting an exam. And so how the product works is that the radiologist can be sitting at their normal workstation, viewing images, and they can simply click a button, and the outputs of the AI algorithm will pop up, where these outputs specifically consist of bounding boxes over detected lesions, as well as what we call a suspicion level, which essentially maps the continuous score of the AI model into a discrete interpretable category. And so with all this, the radiologist is still the final decision maker, but the product is designed to kind of aid in this decision. And so with this product, as has been discussed kind of as a theme throughout this symposium, it's critical to do external validations and tests across different populations. So we've done a number of studies with the product and the algorithm itself, And today I'll briefly touch on one such study. And so it was a reader study, a so-called reader study, where we had a set of radiologists read a set of mammograms in two scenarios. Once using the AI software, and another time not using the AI software, in different reading sessions that were fully balanced and separated in time. And so in this kind of controlled retrospective setting, the idea is to assess how does radiologist performance actually change when they're using the product as intended. And to collect data for this study, we used 9 clinical sites that we'd never used for AI model development or previous testing. And overall, it represented a fairly diverse population. Another theme that keeps coming up is how do you actually evaluate these models? And there's a lot of different ways to look at that. And it's critical to make sure how you evaluate the model aligns with the end clinical use. But as a kind of overall arching message, we saw a significant improvement of performance when the radiologists use the software. And so one way you can look at it is looking at ROC curve. So if we simply plot the average ROC curve for for the readers when they use the software versus when they didn't use the software, overall we saw that the AUC improved. And critically also when you're looking at the binary decision level of whether or not the radiologist recalled the patients, we saw that sensitivity improved 9%, meaning higher cancer detection. And importantly, this improved cancer detection didn't come at a cost of more false positives. So in fact, specificity also slightly increased. And overall, if we look at each individual radiologist and measure their performance in terms of AUC, for instance, we saw that every radiologist improved as well. And though it wasn't a core goal, we did evaluate the model alone on this data. And what we saw is that the model's standalone performance was higher than each of the 18 radiologists when they didn't use the software. But when they did use the software, there's many radiologists that exhibited higher performance than the software or clinician alone. So this kind of points to this idea of hopefully clinician plus AI is better than either alone. And another big table, but so we've done extensive subgroup analysis in this study as well as others, looking at different factors. And one subgroup analysis I'll particularly point out today is looking at performance by race and ethnicity, where we saw that radiologist performance improved in terms of AUC for each subgroup in the study. And for instance, if we look at the two largest subgroups in this population of Black and white patients, we saw that when the radiologists used the AI software, they demonstrated similar performance in terms of AUC, sensitivity, and specificity in these patients. Okay, so we took these results and more extensive standalone testing and a few hundred pages of documentation to the FDA. And we have since gotten FDA clearance for this product, as well as 2 other products. And finally, after this FDA clearance, in terms of clinical integration, so as I mentioned, I was a co-founder of a startup, and in 2020, we were acquired by this company called RadNet, who might be the single largest provider of mammography in the US. And so since then, our team has worked really hard to integrate our software across RadNet, and we've been able to do so where the software is currently processing about 1 million mammograms per year. So altogether, build an algorithm inspired by how radiologists learn to read mammograms. We've done some validation testing, demonstrating generally robust performance. And we've since turned these algorithms into clinical products that are deployed at scale at this point. Okay, then finally, despite all this progress, there's certainly much room for improvement all along this pipeline for mammography and other applications as well. One point is that this pipeline is not completely linear. It doesn't simply stop at clinical integration. It's definitely an iterative process, and it'll be critical to monitor the performance of these models in the real world. And so I will say it's definitely early in those stages, but what we've seen so far has been encouraging. But there's definitely a lot of room for how do you optimally integrate clinician and AI algorithms. And I see that as a big direction, as many others have pointed out going forward. And of course, there's also opportunities to go kind of further down on the workflow, as other speakers have mentioned, of, can we go beyond cancer detection and detection of different diseases to using AI to ultimately help with cancer treatment? And so there's a lot of work being done here that'll be presented, I'm sure, and it's a direction that I'm excited in as well. Okay, with that, all this has been a huge team effort. So definitely want to thank everybody at Deep Health and RadNet, as well as external collaborators. It's been a number of years in the making, and it's been rewarding to work with everyone. And then finally, thank you all for your time today, and happy to take any questions.
Thanks for the nice talk. I have two questions. Because I'm not a computer science guy, I may ask from the biology perspective. But the first question is that how can this model distinguish the cancer region from the normal tissue?
Yeah, it's a good question. And it kind of comes back to this crux of deep learning, where the idea is you collect examples of malignant tissue, for instance, and normal tissue, and then you train the algorithm to distinguish between these two. And so it's essentially trying to craft an algorithm that can learn this process, but ultimately it's based on the data. So you have different examples, and over time, with a lot of examples, it can learn patterns to distinguish between, malignant and benign lesions, for instance.
And the second question is that we know that there are large heterogeneity between different cancer types. So I'm wondering, can this model be transferred easily between different kinds of cancers?
Yeah, good question. I would say it can't directly be used as is for, say, lung cancer, but as Jayashree mentioned, a lot of times the algorithms you develop can be applied and trained on different sources of data. So this issue of, for instance, the needle and haystack nature and dealing with 3D imaging is common throughout medical imaging applications. So certainly I imagine that the algorithms themselves can be applied, but to do so, you definitely need to collect good data to train that algorithm.
Okay, thank you.
Thank you, Bill. I have two questions. One question was related to one of your slides when you're describing your algorithm at slide 47. And I think in that slide, it kind of showed the link between the 2D model and the 3D model. Is the 2D images informing the 3D model? Like, are they connected or are they separate?
Yeah, good question. And short answer is yes. So the idea is to leverage what the 2D model has learned to inform the development of the 3D model. So certainly the 3D kind of slices have a little bit different statistics, but they're not so different that you wouldn't want to, say, start from scratch. And so the idea here is to take the 2D model to generate these so-called MSP images. So the 2D model is run on the slices and essentially extracting the patches and condensing them down into a 2D image. And then from there, we essentially have a 2D problem where we can directly fine tune that 2D model on that data itself.
Okay, thank you. And I think some of your data sets, some of them were weakly labeled and strongly labeled. So these are highly dependent on the 2D strong images?
Yes, this algorithm for sure. Since then, we've collected strong annotations for the 3D data. But yeah, I think if we didn't have any strongly labeled data at all, the amount of data you need to train these type of models kind of expands exponentially.
I see. And one more question. I think at the end, you mentioned that you are excited about incorporating multimodal data. Is there like a data of in your mind that you think it'll be specifically helpful?
Yeah, I think there's a couple of sources of potential data to combine. I mean, certainly you can think about maybe combining pathology information with radiology. So kind of have a microscopic view with a more macroscopic view of the radiology. And so those are directions I'm super excited about, and I think there's avenues there.
Thank you.
Thanks.
Sorry, I'm going to switch to a virtual question really
Yeah.
quick, and then we'll come back to the in-room questions. Is the solution applicable globally? For example, are there the same quality of 2D, 3D images available in all parts of the world?
Yeah, it's a great question. And I think it is one advantage there is in mammography is that there's kind of just several manufacturers and the equipment is, there's less variability in the types of images that are acquired. Certainly there are issues on potential population shifts and so on and so forth. But for instance, we've done some external validation studies where we've tested overseas and for instance, a population in China. And we saw that the performance of the algorithm was consistent of what we see in the US. So that's promising. And it definitely points back to helping improve access. So screening mammography exists mostly in a certain number of countries. And hopefully if the algorithm works for these different populations, it can be maybe provide a way to boost screening in these populations as well.
Thank you. James?
Hi, Bill. So you mentioned you process 1 million images a year. Like, wonder if you could talk a little bit on the economic side of this. Like, there, it seems like there's a lot of computing cycles that probably go into processing all those patches and processing each stage. Like,
yeah,
is it worth it?
Is it worth it? I mean, ask the patient who we help detect. So I think so. I mean, I think, of course, you want to pick a good application and demonstrate that it does help with clinical performance. And so in that sense, I think these are good applications as any to spend the GPU cycles on. And certainly there's a lot of infrastructure that needs to be built to process all these. I would say maybe the most interesting thing is for some of these algorithms, for instance, the 3D data, and it's probably the same thing with huge pathology slides, is you're less GPU limited and you're more like bandwidth limited, kind of pushing these images around. And like a 3D study can be a gig. So if you're processing a million studies of gig a year and trying to push these around, it definitely adds the limitations of like, should you do this in the cloud or should you do this on-prem? I think generally we've seen that on-prem is better for these type of things, but also help hurt scalability.
Bill, great talk. Thank you very much. I have one question, maybe two related questions.
Yeah.
You mentioned that in deployment in the field, radiologist has to push a button.
Yeah.
Have you studied when they push the button? And the related question is, have you done sort of post-marketing research to see how to optimize that?
Yeah, it's a great question. And I think one of the most interesting areas of research going forward is kind of clinician AI interaction and not just trying to have higher standalone performance. And so in terms of when radiologists click the button, as been discussed today, there's a lot of variability. So some radiologists actually prefer to have it pop up at the beginning. Others do their full read, pop it up at the end. And I think, they probably change over time as they get more comfortable with the software itself. In terms of post-market surveillance, we have been starting to do that. It's still early. And fortunately, what we've seen is promise of higher cancer detection rates like we observed in these studies. Thanks.
I know that there are more questions in the room and there's some online as well, but Bill will be here for the remainder of the symposium, so you can ask his questions there. And I'll try and get the virtual questions in front of him as well. So this brings us to our break. So we're going to shorten our break a little bit because we are running behind. So we will rejoin live here in about 10 or 12 minutes. So please enjoy your refreshments. And for our online participants, we will see you back in a little bit. All right, we're going to start again. So if you could find your seats, and welcome back to our virtual attendees as well. Great. So I would like to welcome Dr. Singh to the podium for his talk.
Well, thank you so much for the invitation to present our work. Today I'll talk about how we use microscopy images of cells to make predictions that may help us with right? Finding new drugs. And I know this is a bit of a detour from what you've seen earlier today, but I just noticed Pathology is right next. So sort of would set the stage for that. I'll focus on two main aspects today. Just first on how we extract valuable information from millions of cell images to characterize cell state. And then second, how we utilize that information in biomedical research at large, but more importantly for us in the pursuit of discovering new drugs, new therapies for various diseases. So microscopy images can be a real work of art. You might see some in the next talk as well, but most of you are likely familiar about the beauty. But what's even more amazing is really that the wealth of information that's hidden within it. It's true with medical imaging as well. But if you can sort of unlock this information, which you can do using bioimage analysis software, that's really where a lot of the work that the imaging platform in which my lab is embedded was known for. The imaging platform of the Broad is home to one such software, Cell Profiler. It's a widely used tool that we and others around the world employ as a workhorse for analyzing images. I won't be discussing Cell Profiler at all today in detail, except to just highlight that one of the most rewarding aspects for its creators, which includes Polina, by the way, has been the contribution to the discovery of new therapies for diseases. But today, I'll focus on how our research, which relies on information that's extracted from software like Cell Profiler, can drive biomedical discoveries with the potential to impact patients' lives. On this front, I just wanted to acknowledge our sister lab, led by Ghassemi, which is dedicated to developing the next generation of bioimage analysis software, building on the work that was done in the imaging platform over the years. The lab is focused on incorporating deep learning innovations into this field of microscopy imaging and bioimaging. So that use of imaging, which where we use tools like Cell Profiler to measure very specific things we care about, identify specific phenotypes that we want, is powerful. And I don't have the time to go into too many of those examples, but you can measure individual things from images, but we know that there's more than a single readout or a single cell phenotype that you can get from an image. In fact, within the field of image-based profiling, we assert that by collecting hundreds of features, hundreds of readouts from images that just look like this, that we can capture an impressively expansive view of cell state. Now, we know that to some extent, this is true because, in classic genetic screens, where researchers would introduce random mutations in organisms like fruit flies, observing the same phenotype would mean usually that genes in the same pathway were perturbed, right? So the question is, how do we take that basic concept and just scale it up? And here's how we do it in image-based profiling. This process is pretty standard for most of our projects, so I'll just walk through it slowly, and you'll see the exact same process really being repeated at the back end for all the projects I'll show you. So first, we just have a bunch of different things we want to test, like chemicals or changes to the cell's genes using things like CRISPR and so on. We expose the cells to these perturbations and let them grow for a couple of days. And after that, we use special dyes to mark certain parts of the cell so that we can have them glow and we can sort of observe what's happening inside them. This is called, in this case, the cell painting assay. It's an assay we designed to be intentionally affordable and efficient. So you can just do it for a few dollars for a plate. If you know what a plate is, it's sort of a tray of very small test tubes. I'm sorry if I know there's a very diverse audience. I don't know how to really calibrate the descriptions of things. So please forgive me if something sound overly simplistic in my explanation. So once we have these colorful cell images, we can use computer vision methods like what we saw earlier today to analyze them and just measure different features. We can take all these measurements for each cell in a sample, and that gives us a kind of a fingerprint for the cells, just like you'd have a fingerprint for the activity of their genes when you do gene expression analysis, for example. So this is super helpful for now studying biology in high dimensions, because now we can just turn the research questions that we have into data analysis problems. And problems that we are most interested in is how do we use that to dramatically impact different stages of the drug discovery pipeline as represented in the schematic. So for example, starting at the very top, can we use this approach to identify signatures of disease and then find drugs to revert them? I'll just focus on one example on this front in collaboration with Mikko Taipale's lab in the University of Toronto. We're exploring the idea of using image-based profiling for rare diseases. Now, when you stain a protein, let's say it's just called LZTR1 in this case that we picked up, we can see its localization in a cell, see where it's expressed inside a cell. But if we introduce the mutation in this gene that causes the Noonan syndrome, as it's called, we observe a noticeable change in where it's localized, right? So this is a clear phenotype, so that allows us to then just test for drugs that might potentially revert that phenotype back to the healthy state. Now, in this experiment, you could also, as for a diagnostic purpose, you could actually diagnose this disorder by looking at a human face. It turns out the phenotype for Noonan syndrome is very, very distinctive. But of course, the whole idea here is to really scale this up, right? So Mikko Taipale's lab has generated an image-based profiling data set for over 1,000 Mendelian diseases, or for about 3,500 or so mutant alleles. And initially, Jessica Lacoste in his lab had characterized the localization of these proteins just visually by eye, just inspecting thousands of images and labeling the localization of each as to where it's where in the cell each protein is localized. But now Marzyeh Hagigi in our lab is taking an automated, unbiased approach, examining not just the protein localization, but the morphology at large, and identifying, using those morphological measurements to sort of come up with a unique mutant signature. This way we can get insights into the structural changes in the cell, but also then screen for drugs that can potentially revert that phenotype. What's really exciting is that in the next 4 years or so, we'll be generating data for almost 80,000 human genetic variants, which could offer an enormous potential for understanding genetic disorders and developing targeted therapies for those. So now moving all the way down, can we also use image-based profiling for diagnostics? So here's an example of how the technology can directly benefit patients in need of blood transfusions. The blood stored in banks undergo changes over time that impair its health and the ability to carry oxygen. And it sort of makes it unusable, especially for like life-saving transfusions that can be really, really critical, right? Now, healthy red blood cells, they transform from these smooth concave discs to lumpy round balls and eventually to these swollen dense spheres that you see at the very bottom. Currently, the way experts examine this is just sort of looking for morphological changes in blood samples under a microscope. They calculate a morphology index, which essentially counts the number of cells that fall into different predefined morphological classes. But obviously, as you know, this process can be slow, right? And very subjective, subject to human error. But using deep learning methods to identify the phenotypes, our initial approach was just to train a classifier to categorize cells into these different morphologies. The performance was promising. Systems agreement was with expert annotated ground truth, sort of matched what inter-expert variance was. So that's great, but we didn't want to simply mimic the human design system with its, this arbitrary phenotype bins that have been defined in the field. And instead, we really wanted to come up with a more continuous metric which could be a measure of cell quality and use that to define an index from this. So we employed a so-called weakly supervised deep learning approach. You heard about this before. Mindon and Juan Caicedo trained a model to predict the age of the bag. So just similar to the example we saw previously, where we are looking at a label of an entire image as opposed to a region. Here we just have a label for the entire bag, not for the individual cells in the bag. And that high-level label is not really that interesting in terms of something we want to predict. We always know the age of the bag. It's always, that label is always there. But the idea was that older bags tend to have more degraded cells than newer ones, and that information about what could be more degraded than the other be used to then learn a representation of the cells. So this fully automated approach generates a proxy measure, really, for each cell's quality, which can then be translated into a new index. We call this a self-learned index. It had a stronger correlation with biochemical assays than the index that you could get just by human annotations or even by the automated methods that were scoring cells into individual phenotype bins. So if validated in follow-up work, this could really establish a new standard for assessing blood bag quality. profiling can be a valuable tool in understanding how potential drugs work. By comparing the profiles against just a library of chemicals with known mechanisms, we can gain insights into their mode of action. So just remember, the mode of action refers to the way a drug produces its effect on a biological system, for example, interactions with specific proteins or cell processes. So through this comparison with this known reference library, we can identify matches, and that could reveal something about the drug's probable targets and provide a more comprehensive view of what that drug, or the potential drug, is doing. So one example of that is in this work led by Greg Way from a postdoc in the group. We compared two different high throughput profiling assays: cell painting, which looks at cell morphology, and L1000, which examines the transcriptional state of about 1,000 genes. We tested 1,500 compounds, mostly FDA-approved drugs across different doses, and we found that for about a third of the mechanisms present in this data set, the compounds were significantly tightly clustered. In this specific analysis, we saw that morphology as a readout was more consistently outperformed L1000 transcriptional readouts in terms of the number of mechanisms it could capture. And you can see the orange bars here being taller than the blue, light blue ones in the plot. But when you performed a supervised analysis, we sort of trained classifiers to predict the MOA classes. The results here in this case are a little bit more balanced. The two axes here show a classification metric, the area under the precision recall curve in this case. So as expected, some mechanisms are able to predict some assay. I mean, some mechanisms better predicted in one assay versus the other. So what we saw overall was really good complementarity between these two. Now, so that was just one vignette, but sort of brings me to another paper from last year from Marzyeh in our lab. She provided data sets for various tasks that involve cell painting and L1000 assays, that's morphology and transcriptional readouts. In this case, we curated data from thousands of compounds of genetic perturbations and chemical perturbations, and we aim to find useful ways of sort of translating between these two modalities. So going in one direction, we developed predictive models that could estimate the expression level of individual genes just based on their morphology readouts. And now we've seen a lot of the examples of this coming up now in the last 6 months or so. But this approach, only worked for a small subset of genes. But the important thing here is that now once you have that, you have those readouts for free, just given imaging. So that's really powerful. And this was just based on the baseline logistic regression models that we have. Right? But you could also go in the other direction, where we're building deep learning models to predict what a cell may look like if we already know its transcriptional state. This is sort of related to previous work several years ago, where we synthesized images of cells that were treated with increasing doses of a compound, and we were able to predict the synthetic images of those cells, and they look very similar to what the actual cells look like under a higher dose. And this sort of tool, the same idea, could be used to sort of give us an understanding about the relationship between transcriptional and morphological states of cells, which we can explore more through these simulations. Another application of profiling is to identify gene function by grouping similar genes and alleles. We've tested this idea using genetic perturbations, and we found a remarkable ability to cluster them based on similarity in this work by Mohamed Roban several years ago. By overexpressing each gene in a cell population and then applying cell painting, then extracting profiles using the same workflow I showed you, we can rediscover known biological pathways in a single experiment. For example, the RAS pathway is found up here, the Hippo pathway down here. We also saw meaningful anti-correlations, such as negative versus positive regulators of the same pathway. We also discovered an unexpected relationship between Hippo and NF-kappa B pathways, which was later confirmed in follow-up work. So this was our first foray, really, several years ago, into exploring the complex genetic relationships and pathways using cell painting as the readout. And just related to that, one potential application we're considering, we don't really have very strong evidence for it yet, but it's to predict drug response based on these readouts. So a step towards that would involve, for example, stratifying patients and characterizing the disease alleles of their mutations based on morphological profiling. For example, in lung cancer, the disease can be caused by a variety of underlying mutations, as many of you know. So if you overexpress each disease-associated allele in a cell line and then compare those profiles with each other, you can discover groupings of these alleles into various subtypes. The groupings that we saw here broadly align with known biology. But more interestingly, as we can zoom into a single gene and find groupings within those and see what we can get from that. And this is work led by Juan Caicedo in this paper from last year. Here in this case, we took BRAF, for example, and there are more vignettes, but BRAF, in this case, we saw the similarities on cell painting morphology data of these alleles, the BRAF alleles, mirror what was known about the activity of those alleles. You just overexpress the wild type, overexpress each mutation, and cluster them and compare the groupings with the known biology of those, and found a lot of consistency in the way these groupings were emerging. So what's really exciting here is that morphological profiling isn't really disease specific in any way, right? So we could just run it on every variant discovered in every sequencing study for all diseases, which could help us get a better hold on the diversity of genetic variants across each of these disorders. There's an exciting technology underway that could really scale up these experiments. The Blaney lab at the Broad has developed a technology to do pooled image-based screens. This was a couple of years ago. And we're now working with them and the Neal lab at the Broad to create a pooled version of cell painting. The idea is to develop, to perturb cells using a pooled perturbation library, like a pooled CRISPR library, where different guide RNAs in that target various genes simultaneously, allowing for really large scale gene manipulation in a single experiment. Right? So we then do that, perform cell painting, get per cell image-based profiles. We read out then also the barcode of the guided RNA present in each cell to do so, which will tell you what was the genetic perturbation applied to that cell. And the trick for doing that is you just generate. There's a way of sort of generating many copies of the barcode within the cell and then just sequencing that using in situ sequencing. So you can sort of assign perturbations to each cell. You've got the morphology of each cell. And this can really be a game changer in terms of massively scaling up cell painting genetic screens, like the one I just described in the previous slide. Cell painting also has a great potential in predicting various assays. So running large-scale assays for drug discovery can be really costly and challenging, especially when you're dealing with primary cells or organoids or whole organisms. But a few years ago, an international team, including Janssen and several other collaborators, had shown that a single high-throughput imaging assay can be repurposed to predict the biological activity of compounds in different assays, even those that were examining completely unrelated pathways. Working with Kevin Young from Tommy Yakola and Regina Barzilai's group at MIT, Nikita Moshkov and others in our group combined chemical structure information and transcriptional readouts together with imaging to predict assay outcomes, and this paper should be coming out in a week or so. So the results were really promising overall. It sort of showed that image data from high content assays like what we are using here, also along with other available information, chemical structure or transcriptional readouts, can really be used strategically in a multimodal way to design chemical screens and boost hit rates based on that. Cell painting can also be used to predict cell health markers. Paquita Vasquez and her colleagues at the Broad developed a cell health assay that measures about 70 or so phenotypes related to cell health. These characteristics include the different stages of cell growth, programmed cell death, how quickly cells are multiplying, and many other important indicators that give an insight into the overall cell health. So we gathered data from 3 different cell types, altered specific genes almost at random. And we studied them using both the cell painting assay and the cell health assay. And we used the information from the cell painting assay to predict the cell health indicators. Greg Way and our group showed that we could accurately predict many of these indicators, and that sort of highlighted the utility of using cell painting, not just as a way of profiling, but also being able to predict other biological readouts that one might care about that are not obviously available in morphology. So the assay prediction and virtual screening applications I've discussed, they both greatly benefit from having access to more data. This is why we organized the JUMP Cell Painting Consortium. It's a consortium of about 10 pharma companies, several academic groups, including us at the Broad. Our goal was to create the world's largest publicly available cell painting data set. It currently includes about 150,000 compound profiles and genetic perturbations for more than half the genome, generated across multiple sites. So through this data generation effort, our goal was to tackle various different assay development challenges related to cell painting, but also computational challenges that could really accelerate development in this field. Excitingly, all this data is now publicly available as of a couple of weeks ago, and you can learn more at this website. By the way, the slides are also available online in case you want to dig into any of these links. And you can also browse around this data using these portals that we did recently by 2 companies that allow a more sort of visual exploration of the images, but also of chemical structures in case that's the kind of thing you care about. We think this data set could be a real catalyst in terms of having computational biologists and biostatisticians collaborate on this incredibly rich set of fundamental problems, learning effective representations from images, figuring out how to correct for batch effects, translating signatures across cell types, integrating data modalities, and many more such problems. And one such ongoing effort in our lab is to develop just batch effect correction methods, primarily by adapting successful techniques from the RNA-seq and single-cell RNA-seq world, where there's a lot of activity in the space. Batch effects can seriously obscure biological signal. This might come as a shock to Rafa, for example, especially in high-dimensional biological readouts. Our goal is to create strategies for that not only correct for these batch effects, but really allow us to facilitate data creation across experiments. So this would allow others around the world to build on this resource by adding their own data sets to it. I just wanted to close with a quick preview of our next big project. We are creating a consortium now of more than 30 organizations who've joined us to pioneer an integrated approach for predicting chemical toxicity. The current methods for this, for predicting toxicity, involve animal testing. It's expensive, it has ethical concerns, and it's really bad in terms of its actual accuracy. So the field really needs alternatives. Our consortium here is going to be collecting data for compounds with known liver toxicity, and then profile them using multiplex assays like cell painting. And our goal will be to test if just simple methods like cell painting can be used to predict, at least score higher, compounds that are likely to be toxic to humans. And we think that this could really be one of the most impactful ways of transforming the way a lot of the testing is done in both the agrochemical as well as pharmaceutical industries. All right, I think I'll stop there. That's all I wanted to talk about. Thank you so much for inviting me again. And that's our lab. And thank you.
Yeah, so that's a very interesting talk. So I was just wondering, so I was just wondering about the question RNA. That there was some discrepancy. There was some sort of clustering. All painting better in RNA, but then later, yes, that pick the RNA. Wondering, you're not quite saying problem. I have a question to that effect.
Sure.
It also seems much larger collection of data.
Great. Yeah. No. Thanks for the question. Yes. So what we found is, and obviously the study has more layers to it, but one of the fundamental issues that could be explaining it is just the fact that we were sort of trying to compare what we thought was apples to apples, in the sense that what is the most high throughput way, what is the comparably high throughput way of producing transcriptional state of a cell that could match with cell painting, at least in terms of being somewhere in the same range of the cost. So at the time, at least, L1000 is about 3 times more expensive. Doing RNA-seq, I think, was something like 200 times. I forgot the scale. It's like something astronomical, right? So we had to sort of go with something that is reasonably high throughput. So obviously, that number one, that comes with some with more errors, less accuracy. You're only measuring 1,000 things. So some of it could just be that you're measuring fewer things. It's noisier. It may not be an implication about, it may not implicate gene expression per se, but it does implicate, the ability to collect gene expression at the scale we need. But the other part of it, I guess you're wondering, is there a contradiction with the prediction part? So I think number one, it's just like there are a handful of genes, at least as far as we knew, could be predicted. After 1,000, they get the fraction, but it was in the tens. So you can't predict the entire transcriptional landscape, right? But also that that can vary across cell type. It can be context dependent. So I didn't really see a contradiction there. It sort of, for me, it was more, to the extent that there is an overlap of the two subspaces, you're able to predict one from the other. To the extent there's not, you're able to capture complementary, you're able to sort of read out on complementary pathways in terms of being able to better capture one mode of action versus the other, depending on which assay you have.
Any other questions?
As far as I understand it, this cell painting assay using like a multiplex immunofluorescence, what?
Yeah, these are just regular dyes, chemical dyes like DAPI and
Gotcha.
Yeah, that's right.
And so those, is there like equivalents, those dyes that you might find in like some of the earlier speakers talk, like if you're using multiplex immunofluorescence and you do kind of the similar morphological profiling that you're doing in Cell Painter to get similar results?
Oh, great question. Yeah, so the cell painting assay, the benefit of that, of running that, is really that, number one, it's highly standardized now. There's a company that, maybe two now, that sells cell painting kits. So you have like very, very standardized protocols now. We've got standardized ways of sort of setting a microscope in a way that you can acquire them in a way that's consistent. But having said that, if you're measuring many different things about a cell, you're generally able to sort of capture, you're able to get some sort of a snapshot of its morphological state. And I don't know the right number of stains, but once you achieve about 5 or 6, and as long as they're relatively orthogonal, you're able to get out a really rich description of that. So I guess the short answer is yes, image-based profiling is not equal to cell painting. It could be anything that is reading out many different things about a cell.
All right, thank you very much. And our next speaker is Dr. Beck.
Great. Well, thanks for all the wonderful talks so far today. And I'm excited to give a presentation that builds on a lot of what we've heard about on the power of AI, particularly in medical imaging. And here I'm going to focus on histopathology, which we actually haven't heard a lot about. So pathology is, the area in medicine where tissue is taken out of a patient, sent to the lab, and really is tasked with making the definitive diagnosis. So frequently in radiology, the sort of bottom line will say impression, it's a mammogram, impression malignancy, it's a mammogram, impression totally normal, you don't need anything. So if it's something concerning where there's a next step to get a confirmatory diagnosis, the next step is frequently taking out tissue and sending it to pathology. And because it's so important, the core elements of pathology have innovated fairly slowly because it's so important. You really don't want to mess with it without a lot of confidence that it's going to get better. So classical pathology still remains kind of the by far the most common way it's practiced today. And the typical way a pathology lab works is tissue will be stained with histochemical stains that are very, very old, the H&E, that will morphologically highlight on the order of hundreds of thousands of different cells from dozens of different types on each slide. And then a human is tasked with identifying patterns. Or are there a single cancer cell there? And if there is, what is the subtype of cancer? And then pathology is not only critical in diseases like oncology, but also other diseases like inflammatory bowel disease and liver disease. So I'm not going to go through each of these, but let's just say that for a task that's almost entirely visual and data processing, the deep learning advances, and particularly from about 5 or 6 years ago, are incredibly potentially transformative to the field of pathology. And it takes something that's been very much rooted in kind of history and to make it get better and better with data over time, and to be able to provide single cell resolution analysis of slides where certainly a human by eye you know, it would take a lifetime to analyze a day's worth of slides at single cell resolution, whereas now with algorithms and access to high performance computing and essentially arbitrarily large numbers of GPUs in the cloud, we actually can process images with single cell resolution. And one of the questions I always get is like, well, pathologists are so conservative and they're never going to want to leave their microscope. And that's just really not true. Because, you know, I'm a pathologist. We work with tons of pathologists at PathAI. We have over 400 pathologists across the country who are hooked into our platform. Pathologists really do want to incorporate this technology. And that's kind of my anecdotal observations. This was a pretty nice survey study that just came out where they were asking pathologists, what do you expect? Like, how common will these things be in 2030? And essentially everyone says strongly agree that like digital pathology, deep learning, AI will be totally routine for almost every task done in the pathology lab. But the funny thing is we're so far from that now. So while there's a great sort of enthusiasm and conclusion that this is where things are heading, there's a ton of work to do on the research side, the technology side, the productization, the distribution, and then figuring out effective business models for labs to really make this the new standard of care. So I was in kind of doing some of the very early work in academia, probably now 7 years ago or so, when deep learning sort of first came of age, and started PathAI in 2016. So we're heading into our 8th year. And we've really continued from the beginning to follow this kind of flywheel. And now we have about 700 people. We operate a very large lab based in Memphis. We work with I didn't give my conflicts of interest, but everything you hear is entirely biased by my role at PathAI, as well as we partner with pretty much all of the top 20 pharma companies, where this is becoming more and more routine kind of in the first part of this flywheel, which is using digital pathology, AI-powered pathology to explore new hypotheses. And I'll show some of the technologies that are really enabling new hypothesis generation and evaluation in things like prospective clinical trials. Then with this technology, you can validate new hypotheses in trials, and then to begin to distribute new tests into clinical practice. And then this is a real flywheel because it gets better and better with more and more widespread distribution. So this is what a typical pathology slide looks like. And just keep in mind what you're looking at here. This is a tiny portion of one slide. So a patient who has a resection of a cancer might have, 24 of these slides removed if they're having something big removed. And this is like 10% of one slide. So this is kind of the tasks we're asking humans to do today. It's pretty easy. 10 out of 10 pathologists will say there's invasive cancer here. But what no one will say accurately is, well, how many cancer cells are there, and what are their nuclear characteristics, and how many lymphocytes are there, and how many macrophages are there, and fibroblasts. So we get like zero resolution of anything other than what is the diagnosis. And that was enough when the question was surgery, yes or no. A very binary answer that if it's not super early disease is going to have almost complete agreement. But anything beyond that, just human visual interpretation, that's pretty much the limit. And what we can do with deep learning powered pathology, and this really builds on a lot of what we've seen today, but in a different way. A lot of what we've seen takes very state-of-the-art technologies that are very not widely available and expensive and low throughput. This is essentially the exact opposite. We're starting with H&E stains that have been used for 150 years. Every single cancer patient essentially around the world who's being diagnosed with cancer, solid tumors, is having one of these generated. 100% of clinical trial patients already are being generated these slides. So in a totally non-destructive way, you take something that is prone to aging and other issues, and you take an image of it, which makes it essentially exist, in perpetuity, and then you can do these digital assays on it over and over that will get better year over year. And this is showing one image, an output of a recent model we produced, where overall we've trained models on something like 20 million annotations. So we heard in one of the previous talks that getting supervised data sets is difficult and expensive, and I can certainly say all of that's true. But if you can do it, it enables you to build models in a way that you couldn't otherwise, with really single cell resolution classification of large different sets of cell types as well as tissue regions. So in this example of a cancer, we're showing 5 different sort of canonical cell types: cancer cells, fibroblasts, plasma cells, lymphocytes, and macrophages. and some of the core tissue regions, which now we understand with single cell resolution. So now we can take an entirely unstructured piece of data, the whole slide image, and we can structure it in a few different ways for downstream analyses. One is just these dense high resolution heat maps of cell and tissue regions. So now that can become the input into other deep learning models or other graphical neural nets or types of models that can then discover new spatial relationships between these cell types on very, very large scale. The other thing we can do is we can extract what we call human interpretable features, or HIFs, which is essentially a slide level single summary feature that also can characterize sort of human engineered spatial relationships, densities, ratios within the image. And the advantage of training these very, very high quality, fully supervised cell and tissue classifiers is because it's not too controversial that knowing those cell types and tissue regions is important. So you can really focus downstream analyses on things that we have a high biological prior are important structures within the image. So you can really improve the generalizability and robustness of models while not losing much potential for discovery, because these images have tons of artifact in them. They have many parts of the image are not relevant to subtyping of disease. So by taking as a starting, reducing a whole slide image, which is a bunch of pixels, into dense high resolution heat maps of cells and tissues, we can then extract valuable human interpretable features, but also build new machine learning models directly off of these maps. So the basic questions we're trying to answer are some of the ones we've already heard of today. So within cancer, at least for subtyping of cancer, why are patients not responding to therapy? What are some of the pathologic mechanisms of resistance? Can we better measure existing biomarkers? For example, it's well understood that things like tumor infiltrating lymphocytes are an important feature. It's difficult to measure by eye, difficult to measure scalably or reproducibly. So we can measure a simple feature like that with complete accuracy at very, very high scale. But we can also use these systems to discover new biomarkers in areas where there's no defined biomarker, or to discover a computational novel biomarker that outperforms existing biomarkers. So these are kind of these three use cases we're always thinking about. How do we first improve the measurement of what is already known to be important? Number two, how do we discover new biomarkers that have not yet been discovered? And then three, and this is more of a kind of a practical utility, how do we fully leverage the information in these H&E images, which are on every patient, to prioritize patients who would benefit most from downstream high dimensional molecular testing, which could be something like next generation sequencing, which, while available in many parts of the country, is still widely underutilized, but even more so could be like who would really benefit from, highly multiplexed spatial transcriptomics or something where you don't think you want to do it on every patient, but you may want to do it on certain patients. And we can use the H&E to infer molecular phenotypes that could then be downstream confirmed using other technologies. Here's just some examples. This is from a collaboration with Genentech applying our tumor microenvironment profiling system to a randomized controlled trial of their anti-PD-L1 agent, atezolizumab, in non-small cell lung cancer, where the top performing cluster of features, and here's one of the very simple and top performing features, the ratio of lymphocytes to fibroblasts as a predictive biomarker, which makes perfect biological sense, but it's something that would be very, very difficult to measure by eye because it's the quantitative ratio between two different cell types, but each of which are difficult to manually score. And just from an H&E, we can quantify lymphocyte to fibroblast ratio and see here on the top that patients with high lymphocyte to fibroblast ratio show significant benefit from anti-PD-L1 treatment compared to patients with low lymphocyte to fibroblast ratio who show almost no improvement. And that's just one simple biomarker. And we're doing many studies where we try to look within subsets of patients who are thought to be non-responsive. Are there subsets of those patients with different phenotypes that are predictive of response? So we think there's a ton of information within these H&Es, and part of it's going to be extracting human interpretable features that are known to be important. I mentioned this inference of molecular alterations from the H&E is an important area. Here's one study we published in ovarian cancer where it's been known that aneuploidy is associated with high-risk disease. We found nuclear phenotypes associated with high levels of aneuploidy. And then we showed actually with just the H&E image and just measuring nuclear phenotypes associated with aneuploidy, we could obtain very similar prognostic performance. The advantage of the H&E image is, again, it's available for everyone, whereas only a minority of patients are getting genetic testing done in the US, let alone in the rest of the world. So sort of generalizing this concept, we also recently presented work more broadly showing how you could take a set of hallmarks of cancer defined by RNA signatures and then really just match those up with human interpretable features to really bring together these two worlds, the world of bulk RNA sequencing, where we're trying to infer different hallmarks of cancer based on semantic understanding of the different genes, versus our very deep understanding of these different human interpretable features. And now we can begin to create these maps that show sets of human interpretable features highly correlated in either positive or negative directions with particular types of DNA and RNA alterations. So it's now very routine to get RNA signatures or to perform RNA-seq on patients for translational research or in prospective clinical trials. We think where this is going is we'll do a very similar complementary analysis on the already existing H&E image, which is essentially needed to do before you do NGS to ensure that there's enough tumor there. So there's going to be very rich new data sets coming out. We have a panel of features from the RNA-seq, but a complementary panel of features from the image. And you can imagine you're going to have a lot more confidence in an observation if you see it at both the RNA and the cell and tissue level, versus just seeing something at one or the other. And even seeing aberrations between these two things, which we've seen can indicate a particular mechanism. When two things typically correlate, well, what about in patients where they don't? And here's just one more example of predicting molecular alteration. c-Met is an important biomarker that's very complicated to measure from the DNA level because there's a whole different complex set of c-Met alterations that could in some cases lead to c-Met overexpression. There's no standard of care for how to best measure c-Met to identify patients most likely to benefit from therapy. Here with work that we've done with AbbVie, we show that from an H&E image alone, you can predict with fairly good accuracy patients who are overexpressing c-Met by immunohistochemistry, which is an assay that's not routinely done. And then you can actually gain new biological insights into the components of the tumor and the tumor microenvironment associated with c-Met alterations, which could lead insights into how c-Met is driving oncologic progression in patients. And I think the last example I'm going to give is one that builds on some of the work we saw around tertiary lymphoid structures in an earlier talk today. Tertiary lymphoid structures are another one that are very difficult to measure on an H&E image. So there have been very few really well-powered studies on TLS biology because there's just such disagreement about what a TLS is. Is it defined by morphology? Is it defined by immunohistochemistry? So here we've built a few different types of machine learning models, some to classify types of TLSs and others to classify different cell types within an image. And then we said, well, what if the sort of ground truth for defining a TLS were different RNAs from a TLS-associated RNA signature? So we used graphical neural nets applied to cell and tissue heat maps to predict every RNA within a TLS-associated gene signature. We then clustered patients from only features derived from their image, so the imputed levels of TLS-associated genes into patients with high levels of imputed expression of TLS genes in cluster 1, patients with low levels in cluster 2. And in this first data set that we're showing here have shown that this was a very strong predictor of survival because it's well known that having more TLSs is associated with better response to immunotherapy and better overall prognosis. And then we're able to look inside these predictive models and identify which regions of the image are most salient for making this prediction. And this uncovers regions that are dense in existing TLSs, but also adjacent regions that people by eye wouldn't have manually identified. So we're continuing to validate this on additional data sets. But again, this would be a very powerful tool to be able to start with an H&E and to be able to say, there's cancer here. I know all the different cell types. These are human interpretable features. These are the imputed hallmarks of cancer, and even imputed structures like TLS-associated gene expression. So that's it. And I've got about 3 minutes left. So just in summary, a lot of the work we've done is really built on the big advances in deep learning and computer vision. We've taken very seriously how strong the performance is in fully supervised learning to train models to create these single cell resolution heat maps of cells and tissues. And then with that, we can do a lot of different things. We can better measure what's already known to be important. We can impute molecular alterations, which could build very strong multimodal data sets. And we can also do end-to-end weakly supervised learning on these same images, but now really understand the underlying local features within high attention regions. I haven't shown any of that work today, but there's a lot of really interesting work on combining fully supervised models With weakly supervised models, where the full supervision of knowing cell and tissue makeup allows you to better understand the attention heat maps coming from the weakly supervised approaches. And these sorts of research insights, we think really are going to be the thing that really drives the field forward once this becomes sort of a nice to have to an essential part of evaluating pathology slides. So I'm going to end there and happy to take any questions. Thank you.
Can you give us a little intuition on like how you do that? Like what are the models identifying to help you infer the the transcript?
Sure.
Genomic?
There's two ways we do it, and I'll show you one. One way, and what I showed today, the examples I gave today, which I think is sort of novel. So what people typically do is really stupid and doesn't work very well and is not generalizable, is they get access to TCGA, and they have a bunch of data, and they have a bunch of whole site images, and they put them both in to an end-to-end deep learning model, and they try to make a prediction, right? The reason that's elegant, because it's very easy, but you're so prone to artifact because there's no knowledge. There's not enough priors being kind of restricting where the attention of those models can go to. So that's been like kind of the standard in the field. And we can do approaches like that, but we can do them much more sort of cleverly to restrict what they're doing and hope that they focus on what's important. That allows you to have smaller training sets and more generalizability. That's kind of what we're showing here. And part of it's because we've done enough of the fully end-to-end weakly supervised. And people have been so fooled in TCGA using that approach because there's so much bias in TCGA. There's all these different tumor types, there's all these different sites, there's all these different scanners. So for example, if you try to predict a KRAS mutation from an H&E image across pancreatic cancer, breast cancer, and colorectal cancer, you're going to do very well. And it's going to be because the prevalence of KRAS mutations are very different in those tumor types, resulting in the fact they look different. And it's sort of a trivial task for a pathologist. So I would just say, I'm saying like, that's not what we're doing. Although I think, there are values to that approaches in certain settings. What we're doing here is different. So we're identifying every cell and tissue. And the inputs to that are a labeled region of an image that's saying this is a lymphocyte. So on a training set, you'd start with an image like this, and we actually ask pathologists, we give them jobs and we pay them, and we have 400 pathologists. It's like an Uber for pathology. They're sort of checking their email and they say, oh, a new job's available, and they log on and we say we need 100 more lymphocytes, and they click on 100 lymphocytes. So the training data set is a whole site image that's in a non-exhaustive way has a certain number of regions that are labeled into a certain class. And then the deep learning model is taking as input pixels from the different classes, and we optimize for really good. That's like the simple way we get these heat maps as outputs. So this would be a test set not seen in training. And now these can be deployed at scale. So those are those models. But and then that becomes the input in how you predict molecular changes. And the most elegant thing we've done here is we can use graphical neural nets on top of these heat maps to make the prediction. There's two ways we can do it coming from here. We can reduce into human interpretable features, and then we could do like, lasso or something to make a prediction, or we can just do GNNs on top of these graphs to make the prediction.
So the graphs are kind of identifying like patterns of the cells related to each other. So it's kind of like really spatial. It's not getting at like the morphological cues, like KRAS cells aren't straighter, and that's the kind of feature you're picking up on. It's more that there's certain cells
That's
that are in,
true.
like, patterns.
That's fair. Yeah. I mean, you could have a map that would have nuclear features in space as part of the graph, and then I could learn that. But typically it is just cell identity and location and tissue region.
Super cool. Thanks.
Sure.
Andy, great talk. I had a question about just the end-to-end workflow and where the complexity lies in terms of building systems like this. So, if you were starting today with all the like rapid exponential improvements in vision, AI, do you think that would dramatically simplify what you're building now, or is that just a small component of this whole thing?
Yeah.
For example,
I
just
think-
thinking of
I think-
segment anything that just
Sure.
came
If
out.
I was doing research, I mean, I think there's two parts to the question. I think most people here are interested in research. Yes, it's never been a better time to be doing this. Yeah, I mean, Facebook just released their SAM segment anything, which people are playing around with. There's still issues with having to, I guess, give Facebook all your data to to run it and stuff. So I mean, there's like engineering implementation challenges that I think are still challenging, and a lot of like what we've built is around that. But in terms of like getting stuff to work, yeah, there's never been a better time to be doing this, and it would probably be different if we were starting today. There's certain things we would have gotten to a whole lot faster. That being said, in terms of the actual business and deploying like things for customers, a ton of what we do is like software engineering, scalability, infrastructure, quality, regulatory, interfaces for pathologists, interfaces for pharma companies, that I think are not like, it's just sort of, I mean, there's also been decent advances there, but I'd say the most transformative advances are like if you're doing small projects. And yeah, they're huge, and we should all be using those things, like, because there's great new stuff every day in vision, in language, in ways of integrating multimodal data. So I think it's the most exciting time to be doing this work.
Thank you very much, Dr. Beck. And last but not least, Dr. Golland.
Good afternoon, everybody. So I have the dubious honor of being between you and the exception. Hopefully you'll enjoy the story. My group is at MIT. We're a bunch of computer scientists, electrical engineers, and applied mathematicians working with physicians in the Boston area to solve interesting, important clinical problems where sort of information, clinically actionable information is captured in the images. And the physicians have intuition that if we measure that from images, they can either change how they treat the patient or understand something about the disease that they don't know now, and then ultimately change how they treat the patient. And so what I'd like to do today is show you one of the example and tell you a story of how sort of this research evolved. So this research focuses on heart failure, and in particular when patients this took us a little bit of time to wrap our heads around, but basically when patient's heart is not beating properly, then there is buildup of pressure, and the fluid from the capillaries in the lungs seeps into the air sacs in the lungs. So paradoxically, the easiest way to see the heart failure is to look in the lungs. And so if somebody walks into an emergency room with the shortness of breath, and it sounds like they're running out of breath, the problem usually is in the heart, not in the lungs. And so if somebody comes in and complains about these type of symptoms, the first thing that they get is chest X-ray, in addition to many other measurements. So the picture here is of our collaborator, Stephen Horn, who's an emergency room physician at Beth Israel Hospital, just across the street. I don't know which way to point here to actually point to BI. And so we've been working with him for, I don't know, half dozen years now on this problem. And so what Steve has to do when he sees a patient like that is to integrate all of the knowledge that he has about the patient from blood tests and so on with information in the chest X-ray. And the decision that he has to make is whether to admit the patient to the emergency room, which is the most expensive option. But if he thinks that the patient is really in the critical situation and will recover quickly by giving them diuretics and other medicine, then that's the right solution. Or give them medicine and send them home if it's not such an acute problem, or admit them to the hospital, which would be preferred if you think that this is a problem that is not going to resolve in 24 hours. And this decision is done based on a lot of information, including chest X-ray. And moreover, when the patient is in the hospital and given diuretics, then oftentimes they will get X-ray every 12 to 24 hours. So some of the patients get on the order of 10 X-rays in their hospital stay. And the goal is to understand whether the patient has achieved some steady state or they're dry now and can go home. So chest X-rays basically are used for evaluating the fluid status of the lungs. Okay, great. So now we have all these patients in ICU, ED, hospital with all these chest X-rays. And so there are several challenging problems. One of them is that in the situation of emergency department, there is absolutely no time or resources that would make it reasonable for the treating physician to look through the last 4 visits that this patient had and potential, let's say 30 X-rays to understand the trajectory with how they evolve. And the information from the chest X-rays is not captured anywhere but in the images themselves. So they're not part of the EHR record. So EHR, so as a result, the physician, every time they see the patient, they sort of don't get to refer to the previous information. And the other problem, and so we think it's a problem, and we think we can improve situation by extracting numerical value of how severe the disease is, or this current state is, and providing this to the physician. The other problem is that if somebody introduces new intervention, then assessing that intervention, if the phenotype is the image, you've already seen 6 other examples where basically it's very, very difficult to do analysis if the phenotype is in the image. We really only know how to do analysis on numerical values. And so this is another example where converting it to numerical value would be very important. And because I'm closing this session at this point, I hope that almost everybody here says, well, what's the big deal? Train a neural network, right? Image in, so the image will be analyzed by the neural network, and the neural network will predict, just like everything in medicine, pulmonary edema, which is fluid in the lungs, is evaluated as none, mild, moderate, and severe. Every clinical problem I've encountered in my research has none, mild, moderate, and severe. So we'll predict one of these numbers, and it has to, so at this point, if not you, but your graduate students or postdocs can definitely fire this up, train the neural network, and predict the output. So there is one catch here, which is we don't have a single annotated image. We don't have labels in the language of machine learning. So creative graduate students have realized that we don't have numerical labels, but what we do have is stories that radiologists have written about these images. So when the radiologist is presented with an image, they ask the question and they provide general description of the image as well as they attempt to answer the question. So that's what we have. And so the talk today will talk about how we use the radiology reports to solve this problem where there are no labels. And so our work is grounded in this amazing data set. Somebody already mentioned it. It's MIMIC data set, which was created at BI. It's basically all the patients who went through BI and got chest X-ray in the emergency room or their ICU in the last 10 years, de-identified and released for research purposes. So any one of you who is interested in developing machine learning algorithm for medicine, this is a fantastic data set to work with. And the numbers there show you how big the data set is. It's about 250,000 images in a little bit under 100K patients. So our first attempt at this, which I think is sort of very naive, but we wanted some sort of baseline, is to build a bunch of scripts. We call them natural language tools, but they really are scripts that look for keywords and check for negation, and to attempt to extract labels that way. So for example, if somebody mentions certain finding in the image, then we can assign a certain level of edema based on what the physician, sort of basically there is a table that the physician specify, plus all the synonyms of that same thing. I've discovered that there are about 7 different, like there are these stories about how there are 20 words for snow in Eskimo language. There are about that many words for pulmonary edema in the radiology reports, and like many of them mean the same thing. And so you can do that, and using that, we labeled about 5,000 to 7,000 reports, depending on how confident we are in these labels. And so then you can train the neural network and evaluate it. And I will tell you now, otherwise this talk would be really short. This is not good enough. Basically, the accuracy is so that the physicians look at it and say, eh. And so the rest of the talk will be about what we have done to actually bring it up to the point where the physicians are starting to pay attention. So one thing we realized is that many radiology reports we can't label with these simple tools. And so it's entirely possible if you fed them into GPT, it would be happy to produce some nice poem about this patient, but numerical values we cannot get out of these reports. And there are 2 reasons for that. One of them is that some of the reports are written in reference to the previous image. So some of the reports say no change from the previous time. So some of the research that has been going around sort of in my vicinity, some of it by my students, some of them by students of my colleagues, is to build natural language tools that can actually parse longitudinal text information that does refer to previous time points. And so the other reason, which is actually bigger reason and sort of more radiology reports that we cannot label sit in that bucket, is because the situation is complicated. And so the way the radiologist writes it is that they're trying to convey this to the physicians. And so they're hedging a bit because it's complicated. And so many reports look like that, where you read it and you say, but commit already. And you know that they can't because the situation is complicated and the patient has many things going on. Okay. And so basically our simple scripts can't get at the important labels for training the neural networks. And so here's what we're going to do instead. On the other hand, what we have noticed is that if we take one of the language models, standard language models like BERT that Marzyeh mentioned in the first talk, trained on scientific data or clinical data. At this point, we've tried every version and the performance actually is about the same for every one of these language models. And we use that language model to translate the radiology report into a numerical sort of vector of numbers, right? Numerical representation. Then we can train classifiers that are high 90s accurate at predicting the label. So it turns out that the radiology reports have the information in them. And if you think about this, effectively, so let me point at this. Effectively, here we go. Effectively, rather this radiology report, actually, this picture misrepresents of what's going on. In reality, what happens is we have an incredibly powerful neural network that looks at this image and generates the report. And somehow we keep forgetting about that, right? So this radiology report is an intermediate representation that is already coded in terms that are important to us. And that's why we found that if you put the neural network to work on the radiology report, it's much more accurate than what you can do from images. So it shouldn't be surprising if you think about it for a minute. Okay, and so what we're going to do is exploit that to help the image neural network. And so here's how we're going to do this. The upper stream, let's see if I can talk and point and look at you all at the same time. Okay, so the upper stream looks just like a regular classifier. For now, treat this as a classifier neural network. So ResNet, or whatever your favorite neural network is, right? So as I said, this loss function penalizes errors in labels. And if I do that, it's not good enough. Now I'm going to train a parallel stream where this neural network looks at the radiology report and predicts the label. And these labels are pretty accurate. And so I should say that neither the architecture nor the sort of loss functions for the connoisseurs here, something special. Basically, these are standard architectures. We found that the architecture makes very little difference in this problem. But here's kind of the interesting twist here, I think. We will cut both neural networks somewhere in the middle. And so where to cut is black magic. So you take one layer, you take sort of the middle representation from the neural network that describes the image and the representation from the text. And now I'm going to put another loss function here that effectively will push the representation of the image and the representation of the radiology report that describes that image close to each other in this numerical space that describes them. Okay, so now I'm forcing the text and the image to agree as far as the representation for the neural network goes. So the challenge, of course, is that if you make that without anything else, without worrying about anything else, that there is one way to make it work really well, which is map everybody to 0. Then everybody agrees perfectly with the text, right? But that's not the solution we want. So the real solution that we have sort of invented in parallel with several other groups is to force these representations to be very close to each other if they come from the same imaging event, and to be far away from each other if they come from different events. For those of you who do machine learning, now it's known as contrastive learning, where I will make it close relative to the mismatched images and reports. And so the way we train this neural network is that we take pairs, matched pairs of image and report, and we scramble the order, and those become negative examples. So the positive examples are the matched ones, and the negative examples are the mismatched ones. And we force the representations here to behave as we want, which is for mismatched ones to be far away and for matched ones to be close. And so I think there are two reasons why this should work better. And in the next slide, I'll talk about sort of how big these two effects are. One of them is that this representation of text is already very good for doing classification. And so what happens here is that the representation of text drags the representation of images to align with it. And as a result, the space for images is better organized. So that's one effect. The other effect that we were really hopeful for, but so far it's been a struggle, is that notice that to force this loss function to go down, you don't need labels. All you need is images and reports. And as a result, I don't need to ask I don't need to build an Uber for radiologists, right? I can just take very large data set and sort of intrinsically learn the structure that describes that data by forcing matched things to be close and mismatched things to be far. Okay, and then at deployment, I don't have the radiology report because the patient just walked in and I took the X-ray. I strip away the text part of the model and I just keep the image-based part of the model. And so the evaluations here will be with respect to how well just the imaging part works while I'm training on both images and text. So this is interesting. We sort of didn't understand how the lay of the land was in this area. And we kept talking about multimodal, and everybody assumed that we're going to take the reports. But I'm like, oh no, I can't afford to wait for hours while the radiologist is typing up the report. I want to provide the answer very close to when the imaging happens. So we will evaluate image-only model while it's trained on both types of data. Okay, and so let me tell you, so the papers have many more really boring tables. Let me just kind of tell you qualitatively what we're seeing when we do this. So I have 3 different area under the curve measures. If you prefer to think of accuracy, you're going to have to go look at the paper. But let me assure you, in all of these experiments, accuracy is perfectly correlated with area under the curve. So the number will be slightly different, but actually the relative, which method did better, will not change if you look at accuracy. Okay, so area under the curve for blue, red, and yellow corresponds to those boundaries between 0 and 1, 1 and 2, 3 and 2 and 3, right? So it's none versus mild and everything else, then none and mild versus moderate and severe, and then severe in its own category. I should also say, and so every experiment has 3 bars because basically it's not binary classification, it's actually 3. Okay, I should also say that the yellow bar is always higher, and that's not surprising. In some sense, telling severe patients is much easier than distinguishing the boundaries. And like many things in machine learning for healthcare, that's the least interesting boundary. Because when the patient is in that much trouble, I really don't need machine learning to tell me what to do, or the physicians don't need as much. And the reason why it's so easy is because the scale is not linear. What the physicians are very interested in, and that's why the scale is like that. It's basically sort of log transformed for those of you who think about these things, where it's very important to tell fine gradations when the patient is just starting to be in trouble. Once it becomes severe, basically 3 includes, or severe category includes patients who are just severe or like really, really severe. And those are really easy to tell. And so that's why our accuracy is always, or area under the curve or any other measurement is always much higher at that. And the physicians are entirely unimpressed by it. Okay, so image only is a fully supervised model. I take a neural network, I give it the 5,000 labels, I train the neural network just on images. And remember, I told you the physicians were like, because basically these are the numbers and it's in the 70s. And they're like, when you have it approaching 90s, talk to us. Okay, so jointly supervised is when I do what I showed you there, but with the same labeled data, meaning that it's images and reports, but only for images that I could label with numerical label. So this is the bump from here to there is due to the fact that the radiology reports are dragging the images to be represented like the radiology reports. And the next experiment is what happens when you feed it the hundreds of thousands of images and reports that we couldn't label before with the script. And so this is disappointing. I sort of when we started the research, I was hoping that feeding the thing 250,000 images, like how can it not figure it out? And the answer is we're probably doing it wrong. Like we're doing it in the most naive way, and that's not how we will take advantage of unlabeled data. And it's actually consistent with many other projects where we had great hopes for large amounts of unlabeled data, but it was kind of like a little bump. Okay, so that's where it's at. And that's where it was at when we asked the question, maybe we are doing it wrong. And in particular, one thing that we were doing in this initial phase that was naive is that this numerical representation of the image is of the whole image, and the numerical representation of report is of the whole report. But this is really more like what actually physicians think. Like every sentence in the radiology report says X happened in location Y, or nowhere in the image I observe X. So it has location and thing, right? And so our group and others in the last couple of years have started thinking about how do we build representations of images and text that match this better. And so this is what I'll show you in the next 5 minutes, what we have done here. And the idea is really to build representations that capture here we go. Build representations that capture information just in this window. And I did this by hand. I colored the sentence and the image region by hand here, just so that it's in your face and you can see that it's obvious, right? But hopefully towards the end of the talk, you will see images colored like that by our method. Okay, so what we are going to do is instead of creating one representation, we will chop up the image into smaller regions and extract a whole bunch of smaller representations, and then build aggregate them into what we call an image representation. And similarly, the radiology report will be represented by a bunch of representations. And so these representations are of sentences. So the great thing about radiology reports is that somebody actually told us where the boundaries between units are. Every sentence is sort of a unit. And so we do it on units. And so to us, again, radiology reports are the anchor, and the images is where we have no idea where the information is, right? So the idea is that it gets aggregated here. So basically every sentence gets a description, and every region gets a description, and it gets computed into one image descriptor. And as a result, we have a bunch of scores here. So what do these scores mean? It's how much the sentence describes this image. And then these scores are aggregated. Think of averaging for now. Like we've tried a bunch of things, but I think averaging is good enough here. Okay, and so it gets averaged into an document image score. And it is how much this document, we call it document because, report image score. Okay, and so this is what we used to do basically, where we would find a bunch of features and then average them to create this one vector. And so the new representation creates these region-based representations for image windows cut out of the image and sentence-based representation of reports. And now this aggregator creates basically image patch by sentence collection of scores. And we have investigated different ways to aggregate it into a joint image radiology report score. And it turns out that the right aggregation seems to be where we aggregate at the level of similarity scores rather than features. And again, I'm very happy to talk about more excruciating details about how this is done. One thing I should point out is that for those of you who do machine learning, what we realized doing this work is that my amazing graduate students realized while doing this work is that this problem is related to what in the what about 25 years ago machine learning was really excited about, a field of machine learning was really excited about, and it's called multi-instance learning. So multi-instance learning came about from a problem where, suppose I put some substance into a test tube, so like proteins or something, I shake it up, and if I have some way to measure whether the right phenotype emerged, right? So basically, I get the answer at the level of a tube, not at the level of individual molecule. So for example, if the tube lights up certain color, I know that there are conformations of the protein in that tube that are with the right characteristics. But it doesn't mean that every molecule is in the right conformation. If the tube didn't light up, then I'm guaranteed that every molecule there is in the wrong conformation. And so this is where kind of multi-instance learning was, what multi-instance learning was motivated by. And so we realized that what we are doing is very similar to that. If you think of radiology reports as labels, so a sentence is a label, and I know that somewhere in the image, there is an image patch that corresponds to that label, but I don't know where. Another way to think about it is weakly supervised. So we ended up actually borrowing techniques from multi-instance learning to learn this image patch to sentence matches much better than you could do it globally. And so, as before, the excruciating details and statistics in a paper, I want to show you examples. So these examples are cherry-picked from, so hold on a second. Here are the boring statistics. So this method is basically state-of-the-art in doing this kind of work where you learn matching, you learn alignment between sort of image sentences or image patches and sentences or snippets of text. And here we evaluated on pneumonia rather than edema, but actually we've evaluated it on a whole bunch of pathologies, and the results are extremely consistent, which is that we improve classification accuracy. What do you mean? What do I mean by classification accuracy? So we learn these representations. And then on top of that, we don't start from ImageNet or random weights. We take these representations and we train a classifier using these representations. And so then you can classify images like we classified them in the first one, in the first part of the talk. And we see that with very small amount of labels, we can get to accuracy that compares to using huge thousands of labels. And so this is promising. It means that we are learning something that corresponds to real structure in the images. And as I promised, here are some images. So the images and the text have been cherry-picked by the field as being challenging, and other papers report them as failure cases. So we said, great, this is our test set. So the text here, so this is a task of visual grounding, meaning that the input is text and the output is a heat map that for every pixel says how much the image patch around that pixel is similar to the text or not in this quantitative representation, right? So red corresponds to very similar and blue corresponds to not so much similar. And the boxes are what the humans drew. And so I can convert my heat maps to boxes, and people have been doing that too, but we like to look at the entire heat map to make sure that something sub-threshold doesn't sneak in broadly. And so sort of looking at these examples, I hope you can convince yourself that it's sort of finding things in the right places, which is very exciting because this is basically with like no labels at all. So this opens up interesting possibilities for learning where you don't have an army of physicians to label. Sorry, I want to come back and harp on the point here. You couldn't outsource labeling of this because the technical term, this is not sort of something that you put on Amazon Mechanical Turk and anybody who is bored can just quickly label the data, just like in pathology examples. You need expertise to label, which means it's very, very difficult to do at scale. All right, so before I conclude, I want to thank my fantastic graduate students, amazing collaborators, both at MIT and at Harvard Medical School, and generous funding by anybody who would give us any money. Okay, so to summarize, what I showed you today is an example of the kind of work we do, where we think about the problem and try to come up with computational solutions that help us get information from images. In this particular case, I showed you an example where taking advantage of available text information from radiology reports allows us to overcome this challenge that there are no labels that are reliable that we can get for this application. And moreover, we saw that learning over these joint representations leads to much better performance in the modality specific situations where you only have images. And finally, these local level representations that have very interesting connection to other areas of machine learning improve the performance even more and open up sort of these ideas for how we can support clinical decisions even better in the future. Thank you. So the question is, why not sort of score the severity with a number between 0 and 1 instead of these categorical answers? And the answer is no reason. You should do a number from 0 to 1, and we have models that run like that. They provide no improvement whatsoever over categorical, and the categorical ones, so this is sort of a classical observation in machine learning. The categorical ones are much easier to train. They're much less fickle to like setups and all that. Regression models are just like tiny bit fragile and harder to get to train, but the answer is about the same. Oh, that's a whole other can of worms. So how sure you are is an active area of research in the field, including my group. And basically right now, we don't have anything that gives you anything that I would be happy to sign off on. It's a very challenging problem to actually estimate uncertainty precisely. But just because I gave you 0.9, it doesn't mean that it's actually 0.9. It just means that I'm foolishly confident. The other line of work that we have done is actually something that it's sort of model interpretability. So rather than saying I'm confident, we have done work where I will produce findings that support my decision. And so what I found interesting is that the physicians find that so the way sort of machine learning people think is that it's a way to convince the physicians that your model is right. But the way I think of it is that it's the way to reassure a physician when your model is wrong, because actually findings are much easier to check. And if the physician sees that my model got the findings wrong, then it's sort of reassuring that it got, well, of course it got the final result wrong, because it's wrong at the elementary level. So some level of sort of, I don't want to say transparency, but some low level of explanation we found we can do with the more acceptable accuracy these days. Basic uncertainty estimate, I just don't know how to do well.
I was wondering, did this work spark any ideas on what you think might be a general strategy for multimodal data integration? I mean, here you had just X-rays and reports, but you know, you could potentially get everything about a patient.
That's right.
Are there any general principles you think you could extract from this?
We find that actually quite a lot of what we have done doesn't rely on the specifics of the domain. We find that images and text are the hardest modalities to work with. If you already have tabular data that is numeric, some of the challenges go away a little bit, but some of the principles that we have seen, so one of them is like this local versus global, definitely true about many modalities, that it seems that it's better in general to work on local representations that you integrate at the level of scores that is of similarity rather than integrating the features themselves. Having said that, actually the solution that does best combines the two, which is sort of consistent with what Bill was showing.
I was just wondering, like, especially once you have multiple modalities, doesn't that become a combinatorial problem in terms of?
Yes.
Okay. And so how did you address that here? Or did you just do all pairs of document where you had sort of regions of interest and sentences? You just did all pairs?
So it's, if you committed to N by M features, N features for the image and M features for the text, it becomes quadratic, and that, like N times M. Yeah. Yeah, so in that regard, we don't have much to add besides, yeah. Yeah, at this point, we're not trying to be efficient. We're just trying to get the performance up. Right. So this is an excellent question that actually gets at something different, which is that if you think about this, we're training from either noisy labels that we've extracted or radiology reports that can be thought of as noisy labels. And so how much of the error is in the fact that we are training from noisy labels? And I think folded in that is also if you had two good radiologists and you asked them to score these images, how much would they agree? I think it's always underlying that. So to answer questions like that we had, we ran the following experiment. We took on the order of 200 images that we picked to represent diversity of severity based on our model assessment, as well as like for 200 images, we could just read the reports and say, oh, the radiologist who wrote it thought that this was mild case or something like that, right? So we picked about 200 cases and had 4 radiologists score them, because 200 we could beg them to do and they would do it, like 6,000, forget it. Anyway, so we had about 200 images and we asked all 4 of them, and the majority of disagreement was by 1. But in cases where it was more than by 1, we forced them to talk about the case, and they could resolve some of these, and some of these they could not. So we have an order of 150 images that span the whole severity range that were basically agreed upon by 4 radiologists. And so what we find is that our agreement with the consensus labeling on the 150 images is better than the agreement between the individual radiologists who shall remain nameless because we don't know who wrote the report. But if you look at the report and sort of assign a score to the best of our ability and compare it to consensus labeling, the classifier is more consistent with consensus labeling than individual radiologists. We didn't image anyone. This is what happens in the hospital as part of the clinical routine. So this is not a study, right? This is basically everybody who went through BI, emergency room or ICU and needed an X-ray. And so patients who have heart failure, it's a chronic condition. They go into the hospital once a year and then every 6 months, and then it gets more frequent as the condition degenerates. And so patients who came in repeatedly have upwards of 20, 30 scans. Temporal information. Yes, you could. I agree. So doing it by frame, sort of time frame, as if this is the only time you've seen the patient, is a wrong way to go about. Sorry, it's a very naive way to go about it. And the right thing is to treat temporal information. I should also say, like, I will tell you what you should tell me. Which is predicting pulmonary edema is kind of like an uninteresting question in the sense that it's very hard to act on it. So what happened to us in this research is that to write academic papers, it's great to predict pulmonary edema, but to actually affect healthcare, you have to predict something that the doctors can act on. If I say it's a pulmonary edema of 2 or 3, so what we realized as we actually build these systems and try to evaluate them is that what you should predict is something that can change how the physician will make the next step. And we converged on risk of readmission. So it turns out it's exactly the same architecture, but now instead of predicting severity of edema, I'm going to predict if I release the patient today, will they come back within 7 days or will they come back within 30 days? And these 2 markers, for those of you who are sort of interested in how hospitals work, hospitals really care about because they get penalized dramatically, financially, like, and reputationally, if they have too high of readmission rate. If it's higher than a certain amount, then all of the billing to Medicare next year gets discounted by something like 7%. So it's fairly drastic. And so, but the underlying technology is exactly the same. It's just a different outcome that you're predicting. And so what happened was once we develop the initial stage, we're an academic lab. It's very hard for us to start sort of hardening and productizing things. That's why we have startup culture, right? And so graduate student who actually did the initial part of the work spun out a company after graduation. And this is exactly what they're doing in the company. They're predicting risk of readmission, risk of adverse effects, and they're getting some traction. They're now working. So this was with Beth Israel Hospital, but they're working with VA. So suddenly they're talking about 3 million patients. And they're doing exactly temporal modeling. And so there's lots of interesting questions there because there's temporal modeling at the level of one visit, which could be, for example, 6 days. And there's temporal modeling across visits, which could be like 6 months apart. And clearly the patient's state changes much more between the visits than if you are imaging them every 12 hours. So there are lots of interesting methodological questions of how do you model this hierarchical structure of changes. Lots of interesting stuff to work on. Yeah, excellent question. Thank you.
So to close us out today, Dr. Ree will a short statement.
Hey everyone, I'm Martin Marzyeh. My main job today is to close us out and to thank everyone. So first of all, thanks so much to all the speakers. This was really great. I think kind of what I was hoping for in this kind of meeting. I learned a lot of things, got some new connections and ideas. Hopefully our participants also got some new ideas and maybe some new connections. Thank you to everyone who participated and listened in online and in person. And then finally, probably the most important, I'd like to thank the actual real organizers who pulled this whole thing off. You met Nina and Susie at the front there, and you've seen what Erica has done today. She's led us through this, and she put together everything else that led up to today. So thank you very much, Erica and everyone else. Thank you. Okay, super. And with that, I think it's time for our reception. Looks like it's been set up in the back. So yeah, thank you. Thanks, everyone.